Good morning to all of you. Sir, can you please confirm I'm audible to you? And Dr. Tushar Gupta, sir.
Yep, we can hear you.
Sir, I'm audible.
Yes, sir. Yes, sir.
Can I start?
सिस्टम क्या
सर ये ऑनलाइन कनेक्ट किया हुआ है यूट्यूब के लिए।
नहीं, बट इनको क्यों आ रही है? चलो स्टार्ट करो। यस प्लीज।
Charan, you can mute the another system, otherwise we'll get the echo.
Sir, I have already
Mute in the sense you can reduce down the volume of it so that voice will not come from that system. You can put it mute, but also your mic and also volume be just make it volume. These online meetings are always the same, right? There's always some or the other technical difficulty. So that's a part and parcel of the technology.
Yeah.
Now Charanji is okay, I think.
Is it fine, sir? No.
Yes.
परनीत जी, आप अपने सिस्टम की आवाज को चेक करो। ये उसमें एक और कोई तो नहीं चल रहा है। सारे मोबाइल, किसी भी मोबाइल या किसी से ज्वाइन ना करें।
Birla Institute of Technology and Science, PETS, Planning. So he obtained his B Pharmacy degree from UIPS Punjab University, Chandigarh in 2004, and following which he did MS Pharm in Pharmaceutics from the National Institute of Pharmaceutical Education and Research, NAIPER, Mohali. Further, he obtained his PhD in Pharmaceutical Sciences from NAIPER, and he was an exchange research scholar at the University of NAAC Health Science Center, Memphis, for 1 year. After that, he did his postdoctoral training at the University of Nebraska Medical Center, Omaha, USA, in the area of miRNA therapeutics for pancreatic cancer in the lab of Professor Moga. His research interests include the nano-based delivery systems for all small molecules, microRNAs, and CRISPR, genome editing tools, wherein his lab is involved in designing biodegradable lipopolymers grafted with varying pendant drops to impart multifunctionality. In addition, these lipopolymers are being investigated as carriers for gene and protein-based therapeutics. His lab is well funded from various government agencies, including Department of Biotechnology, Department of Science and Technology, and ICMR, and pharmaceutical industries, to name a few. He has published over 55 peer-reviewed publications, edited one book, and currently is editing a theme issue of Journal of Control release on brain targeting. Further, he have filed 6 Indian patents and 1 international PCT patent application, out of which 3 Indian patent applications has been granted. He has also founded a nanotechnology-based startup, that is Nanobread Innovation Private Limited, incubated at Planning Innovations and Entrepreneurship Development Society at PETS Planning, to further take up the commercialization activities. So today he'll be speaking on the application of drug delivery in the brain tumor by taking some of the small molecules into consideration. So I invite Deepak sir to have a talk on the same topic. Over to you, sir.
Yeah, thank you, Dr. Charan. So I share the presentation from this side.
यस प्लीज चरण जी ये अपना बंद कर लें, फिर डॉक्टर साहब शेयर कर लेंगे.
Okay, so are my slides visible?
Yep.
Okay. So thank you, Dr. Charan, for giving the introduction and setting the base. And I'll first take this opportunity to thank Ibro and the organizing team of ISF College of Pharmacy Moga for giving an opportunity to present the work that my lab is doing in the area of brain tumors, particularly Director Sir, Dr. G.D. Gupta, and then Professor Nagan, Dr. Charan, Dr. Ahmed, and who all are instrumental in organizing this whole activity. Great thanks to all of you. Coming over to the topic, so the topic which I will be discussing today is on the designing certain systems for the delivery of chemodynamite. So this particular molecule is used as a first-line treatment for glioblastoma multiforme and is having a lot of challenges. So, but we will be discussing on the polymeric systems that have been used for delivering this molecule that we have designed in the lab, and also the fatty acid-based conjugates. Now, to start with, what we are doing over here at BITS Pilani, the nanomedicine and gene delivery lab. So the whole area in which my lab is working in the nanocarriers, particularly we are focusing in the polymeric and lipidic system. Dr. Charan, can you please mute yourself?
Hello.
Dr. Charan, can you please mute yourself? Yeah, thanks. So my lab is basically working in the area of nanocarriers, where we have been designing different polymeric and lipidic systems, and also the hybrid systems, taking the advantage of both the polymeric and lipidic in a single system as hybrid system to deliver the small molecule drugs, microRNA therapeutics, and CRISPR-Cas based systems, particularly the ribonucleoproteins, for genome editing purpose. Now, when we talk about the polymers, right? So the polymers what we are working upon are the polyesters and the polycarbonates, because they provide ample opportunity of doing the modifications. So what we will be doing, we are designing over here different polymers, rather starting from the monomers itself. So we are designing different monomers that could, which we can use for ring opening polymerization to obtain the polycarbonate based polymers, and then further they could be conjugated with different functionalities, so that we can impart certain suitable properties into this system which are useful for the drug delivery application. Say putting up a cholesterol molecule that could impart better uptake, putting up say morpholino group giving better endosomal escape capabilities, and likewise. And also we are working on the lipidic sides, where we are using certain lipids and modifying those lipids also, and then combining these systems to deliver different therapeutic agents. As I said, small molecules, microRNAs, and CRISPR-Cas based systems. And these systems, since they provide, or rather they pose different challenges which are unique to these molecules or therapeutic agents, so we design the systems as per the we tailor-made for particular delivery applications. And also we are working extensively in the area of drug conjugates, where we are using both the polymeric systems as well as the lipidic system to conjugate the drug molecules to enhance their properties. So with the overall goal that would be able to deliver these systems and convert these therapeutic agents with better safety profiles, better efficacy profiles, and deliver them to the bedside, to the patient. So that is the whole goal of the lab. So the first thing, why nanotechnology, and why we look into this aspect? Now, most of us, we know the advantages which nanotechnology systems give, and majorly being from the science side, being from the scientific perspective, we focus on the clinical need of any molecule, be it a small molecule or a microRNA or any other genome editing tool, if you want to deliver it. That what are the advantages these systems can give us, what are the bottlenecks in those systems, and how we can enhance them. And particularly when we talk about enhancing the properties of a system, we first look at the safety profile. Say if we are having a molecule, now is it possible to improve the safety profile by converting it into a nano form? That is the first thing what we look at. If there is an improvement, then definitely there is a clinical need for doing it. Another part is on the efficacy side, that would it be possible to improve the efficacy of the known molecules or the new molecules also by using the nanotechnology based approaches. And as we all know, there are ample of literature in this area as well, where targeting has been done, both passive and active targeting, that has improved or have shown that these molecules, these carrier systems indeed increase the efficacy of the molecules. And also the third part is the compliance, because when we are giving it to the patient also should accept that formulation and is ready to take it. Say if it is for a chronic therapy, then he has to take that medication for a long term. So there should be a compliance also. So the overall goal to identify the clinical need is to assess the safety, the efficacy, and the compliance. And any one of these, if achieved, if we are performing better than the conventional formulation, then obviously there is a chance for that type of a nano system to go in further. At the same time, which most of the things we miss out is to look at the potential of those molecules and what are the technical and scientific advancements which we are bringing in. Address a particular need. Otherwise, making those formulations and bringing new systems will not be of any use if they don't reach the market. Plus, what are the technical and the scientific inputs which are required and how we are improving that, and any advancements which are coming up. Right, say if we talk about the nano systems, now we can see now a good number of nano carrier systems that have taken place in the field. designing certain, the environment system, pH sensitive system. So these all advantages could be, achieved by using a nanomedicine approach and making it a lab scale. But that size changes when we are producing at a larger scale, and also that size is not, uniform from batch to batch. So those are the certain issues which are there. Then stability of these nano systems, as we all tend to aggregate because of the smaller size. And finally, the cost effectiveness, because if you make the system highly costly, then definitely their utilization will be minimal, and it may not be reach the entire population. So these bottlenecks also needs to be looked into. And in our lab, so basically we try to address all these issues when we are designing different systems. So in today's talk, so I'll be majorly focusing on the area of brain tumors, in which my lab is extensively working, and designing different conjugates for the molecule temozolomide, which is one of the first line therapy for glioblastoma multiforme. Now, as we know, glioblastoma multiforme is one of the most prevalent and deadly brain tumors. It is having an overall months, and it's malignant form. It's very aggressive. Now, there are certain challenges when we talk about the treatment for glioblastoma multiforme. That is basically related to the heterogeneity of the tumor cells. And if we are taking pathway blockers or a single molecule, that may not be useful in such a case because of the heterogeneity, cellular heterogeneity. And then there is a diffuse infiltrate surgical dissection of the tumors, and also the location. And when we talk about from the delivery perspective, this is something that worries a lot, because the tumor is located beyond the blood, after the blood-brain barrier. So any molecule which we are using for glioblastoma multiforme, first it has to cross that blood-brain barrier, and then has to go to the tumor environment that also cross to that blood-brain tumor barrier. So these are the two barriers that it has to cross to reach the cells. So there are challenges which are associated particularly with the treatment or the delivery for the glioblastoma multiforme. Now, the molecules such as lomustine, carmustine, they have been initially approved for the treatment of glioblastoma multiforme, and after that therapy, and later on it was approved as a first line therapy along with the surgical and the radiotherapy. So this molecule has demonstrated a promising effect for malignant gliomas, and it is a second generation immunotetrazine DNA alkylating prodrug. So we'll see what are the challenges which are associated with this molecule and how these challenges could be addressed. So first we look at the timeline for the temozolomide. So it was first discovered in the year 1992. So this is the structure of that molecule. It was discovered by Malcolm Stevens and his team. And basically all the things related to this molecule were first tested, then it entered the clinical trials, the phase 1 clinical trials, then later on it was licensed in 1999 for the treatment of glioblastoma. It is initially used as a second line treatment. And then in March 15, 2005, it was approved along with radiotherapy for the glioblastoma multiforme in newly diagnosed adult patients. In 2008, it was declared as a blockbuster drug, which is having the around $1.02 billion sales. Another formulation, initially this basically temozolomide is available and it is given via oral route. Its intravenous formulation has also been approved by FDA in 2009. Now these are related to the molecule, how it goes. Later on, what has been also seen that the resistance starts coming in the temozolomide therapy, which is particularly related to a particular enzyme. It's a methyltransferase enzyme, MGMT. That basically reverses the effect of temozolomide, resulting in its resistance. So efforts started, people started putting their efforts in reversing this resistance. So can we use MGMT inhibitors? Those have been used then, along with the combination therapies. Plus, people also started thinking that can we use another approaches, say nanotechnology based approaches to deliver these molecules. So then ample of literature is available where the lipidic nanoparticles, nanostructure lipidic carriers, liposomes, they have been prepared, then active targeting also have been achieved, and also the co-delivery. Say combining temozolomide with another molecules like passive paclitaxel, docetaxel, or combining it with siRNA. So all those strategies have been done, and they are being continued also to improve the efficacy of this particular drug molecule. Now, if you look at the mechanism of this molecule and what are the challenges which are associated with this. Now, at this stage, we know that this is one of the promising molecule used as a first-line treatment. So, this temozolomide, although it is having 100% bioavailability when it is given via oral route, but only 10 to 20% reaches the brain. So, after crossing the blood-brain barrier, it has to reach to the tumor, and that happens via diffusion process. So an equilibrium is attained, and it is via diffusion, it goes to the tumor cells. So once in the tumor cells, it is uptaken by the tumor cells. So there it is converted to MTIC, then AIC, and this AIC releases this methyl diazonium ion. So basically, it is this particular methyl diazonium ion which shows the activity. So it methylates the guanine residues on the DNA, resulting in the DNA double strand break, the cell cycle arrest. So those things happen because of the methylation happening. Now this is how the molecule acts. But if you look at the challenges which are associated with this molecule is highly unstable at physiological pH, say at pH 7.4. This change from temozolomide to MTIC happens. It may happen inside the cell when it is reached the tumor compartment or the tumor cell, but it also happens when it is in the plasma itself. So once it is converted to MTIC, then to AIC, finally to methyl diazonium ion. Now these products of temozolomide, they are having very poor permeability across the blood-brain barrier. So they do not go to the blood, go to the brain. So that is one challenge which is regarding the stability of this molecule, and it is having a T-half of around 1.83 hours. So if you look at the limitations, it is having a rapid metabolism and a short half-life, and only 10 to 20% of the administered dose reaches to the brain. And again, it undergoes equilibrium that further reduces the amount of the drug that is reaching the tumor compartment. Another challenge is regarding the resistance which happens with this particular molecule. So there are various, reports that have shown that, different mechanisms have been given for the resistance to this particular therapy. One among them is the MGMT. Now, MGMT is a enzyme. It's a methyl transferase enzyme that basically demethylate the guanine residues. So temozolomide, what it is doing, it is methylating the guanine residues or the adenine residues. This MGMT reverses that. So what has been seen that in temozolomide therapy, this particular enzyme is upregulated, thus resulting in the temozolomide resistance. That may be related to the epigenetic mechanisms which are involved, because the promoter region of the MGMT may get hypermethylated, resulting in the upregulation of this particular enzyme. Also, the tumor microenvironment, the hypoxic conditions of the tumor microenvironment also can lead to the resistance. And there are other cellular mechanisms, particularly the efflux mechanisms that are all, this TMZ is also prone to the efflux that is leading to the resistance. So the challenges, if you look at this molecule from the clinical perspective, is the short half-life associated with them, the rapid metabolism, and the resistance. Now the question comes that, is it possible to improve the therapy by adopting certain new strategies by modifying the temozolomide, maybe converting it into a carrier system, or loading it into a carrier system, or modifying it by conjugating it. So there are approaches which are there. Now, for example, studies have been done where liposomes, solid lipid nanoparticles, dendrimers, these polymeric nanoparticles, they have been used. And we all know that these systems provide advantages in terms of crossing the blood-brain barrier, then having targeting so that they can target that particular tumor cells. So all those advantages could be obtained from these systems. But still there are challenges when we are using these nano-based systems. Now this particular molecule is having a solubility of 5 milligram per ml. Now we all know that when we are physically loading this molecule with such a high water solubility of 5 mg per ml, it is always difficult to load in these carrier systems at appreciable drug loading. And another challenge is on the release side. The drug tends to come very fast from these carrier systems. Say if I design a polymeric nanoparticle, I may be able to load it efficiently, but the release is also faster from these systems. So these are the challenges which are associated. So what we thought of, can we make the conjugates for this molecule? Now, two options. We can go with the polymeric systems to design the conjugates, and also we can go with the small molecules. So there have been reports for designing conjugates for these as well, and it has been shown that there is an improvement in the stability of the temozolomide. We know that this molecule is having the rapid metabolism and degrades very fast when it goes to the physiological pH. So there is an improvement which has been seen. Also, these systems can provide a sustained release. We can increase their circulation time and hence their chances to go to the brain. Also, by modifications on these molecules, the blood-brain barrier permeability could also be increased alongside the targeting and the cellular uptake that could also be modulated by modifying the systems. So all these advantages could be obtained from the conjugation approach. So initially, I mean, there have been papers like, for example, Patel et al. They have demonstrated that this particular molecule, temozolomide, could be conjugated to polymalic acid. So there they have demonstrated that there is an increase in the half-life of the molecule. The efficacy is also improved. So they have done all sort of tests to prove that the conjugates are showing very, very good activity, as well as the approach is beneficial. Apart from that, conjugates with chitosan, they have also been prepared. Like in this case, they have obtained a 17% loading when they are conjugating it. In case of chitosan, the report is they reported around 4.9% the loading of the molecule in the conjugate. So now we know that these polymer conjugates could be made and they could be utilized for the therapeutic purpose for delivering this molecule. Apart from that, we have ample literature, as well as we have worked in the area of designing these polycarbonate based systems and use them extensively for drug delivery and gene delivery applications, where we have designed these polycarbonate based systems and then modified those systems. The advantage these polycarbonates give is the ample opportunity of modification and grafting that could be done. So this part we have already established in the lab on the synthesis side, right, starting from the monomers and then modifying these systems. Now, if you look at a polycarbonate, so we can start from this particular monomer known as bis MPa, and cyclization for this could be done to get a cyclic carbonate monomers, following which we can do the ring opening polymerization. So after that, this particular group, the COH group, for initially it was protected, then it was deprotected, so that we can get the carboxylic acid groups on the chain itself. Now, these monomers, either these could be modified, could be functionalized, or after synthesizing the polymers, we can functionalize or attach the molecule of our interest into the polycarbonate backbone, and could be used for a variety of applications, for targeted delivery, for designing conjugates, for nucleic acid delivery, designing bioactive polymers, doing for bioimaging applications. So these polycarbonates have been extensively explored, and we have basically covered the complete area that where has been used, what are different applications, biomedical applications in this particular article. On this monomer 2,2-bis hydroxymethyl propionic acid, that how it could be used and further modified for various applications. Now, that is on the polymer side that we selected to go with the polycarbonate approach. Now, on the small molecule side, so there have been reports wherein temozolomide has been conjugated with small molecule, like in this case, PATEL alcohol has been used to conjugate temozolomide for the treatment of resistant gliomas, where it has been clearly shown that after the conjugation, there is a reduction in the IC50 values for in different cell lines. The efficacy has been improved. In our lab also, we have tried various fatty acid based conjugates to conjugate small molecules to improve their therapeutic efficacy. Like if we are having a fatty acid conjugate, so it's basically the chemistry part, that how we can modify the drug molecule, because there should be a group on that drug molecule which needs to be conjugated with either a polymeric molecule or either with a fatty acid, so that it can form a prodrug, and again when it goes to the circulation, we'll be able to get the bioactive form of it. And also these fatty acids, they could be able to enhance its properties, enhance its stability, also permeability. So these are basically the requirements when these fatty acids or fatty alcohols have been used. So there are again many reports are there where fatty acid systems have been used for delivering the different therapeutic molecules. And among them also, people have shown that conjugating, say, oleic acid or linoleic acid or palmitic acid has improved the therapeutic benefit. So this sets the base for designing different systems. So can we design the polymeric or the small molecule conjugates of temozolomide? Now we know that this is the structure for the temozolomide, and it is having a CONH2 group. So in order to conjugate this molecule, we need to have some active groups and some functional group that could be conjugated, right? So by doing the oxidation, we'll be able to convert it into its carboxylic acid derivative. So having COOH group on this molecule. Now this again could be conjugated to a polymer. So this polymer was designed in the lab, which is having a carboxylic acid pendant group. So this particular temozolomide with COH is further modified to have hydrazine. So it is Bohr protected, then reacted with hydrazine hydrate to get the hydrazine, temozolomide hydrazine derivative, and that is conjugated to obtain the polymeric system. Now this polymeric system is basically amphiphilic system that contains polyethylene glycol chain as well as the polycarbonate backbone, so that this could self-assemble to form a micellar kind of a system and could be used further for the delivery application. Again, having temozolomide, since we are having a COH group, this could also be reacted with another fatty acid, but fatty acid again has a COH group, so this needs to be modified to obtain the NH2 group, so that we will be able to couple temozolomide with that fatty acid to get the conjugate. So both these approaches have been tried in the lab. So in the first approach, where polymer has been conjugated, what we have seen that we designed this polymer, it's a multi-step synthesis process which has been used right from the synthesis of the monomer. A carbonate-based monomer having a benzyl group was first taken, then it was synthesized using 3 to 4 step synthesis, followed by ring opening polymerization along with MPEG to form this particular polymer. The polymer was reduced to obtain the carboxylic acid groups, that is on the one side. On the other side, temozolomide was converted to its carboxylic acid derivative, followed by its hydrazine derivative, and then reacted with the polymer to obtain this entire conjugate, which is having a molecular weight of approximately 22 kDa with around 38 units of temozolomide attached to it, which basically corresponds to around 28% drug loading. So the polymer is thoroughly characterized using the 1H NMR, which is shown over here, then carbon-13 NMR. So all these studies, all these characterizations for this polymers as well as all the monomers and the intermediates were done. That shows the successful synthesis of this polymer. Then after forming this polymer, we went ahead for the testing part, for formulation part. So the nano formulation was formed that showed a particle size, the size is of around 180 nanometers, and we can show in this particular same image that these particles were spherical. So we have seen that these, once we formulate this, we are getting the nano-sized particles which could be good for the delivery application. Now, whether there is an improvement in the efficacy or whether we were able to get similar efficacy as that of the free temozolomide. So we went ahead for the cell culture assessment where C6 glioma cell line, so it's a red glioma cell line, was taken to test and compare the efficacy, where an improvement in the IC50 was observed in this particular cell line. And also in the apoptosis assay, we have seen that both temozolomide and PDC, they were showing similar apoptotic activity. Now, one of the challenges that we have seen is on the stability side, that earlier I've highlighted that temozolomide is highly unstable. So after conjugating, whether there is an improvement in the stability or not. So this particular graph shows the particle size stability. Say we are making a nano formulation, it has to be stable on bench. And also the temozolomide stability, both we need to consider. So the first graph shows the stability data for the nano formulation that has been made at different pH, the pH 5, 7.4, and in milk water in which it is prepared. So what we have seen that at the lower pH, say if we are taking pH 5, then it is stable, and at milk condition also in milk water, which usually has a pH of around 6 or 6.5, there also the particles were found to be quite stable. And if you look on the stability of the temozolomide, so a UV based assay was developed to assess the temozolomide stability, and again at different pH. So if you look at the temozolomide, which is kept at pH 7.4, we can see that it immediately degrades. This shows a faster and rapid degradation to its inactive metabolites, MTIC and then AIC. But on conjugation, this is a graph for the conjugation kept at 37 degrees centigrade, again pH 7.4, that showed a marked improvement in the stability. Now, if you look at the other pH at pH 5, at 4 degrees centigrade temperature, pH 7.4 at 4 degree centigrade temperature, the drugs are quite stable. But what worries us is at the 7.4, because it is the physiological pH and at 37 degrees centigrade, which the data clearly shows that there is a marked improvement on the stability side for temozolomide. Now, another aspect, so this is for the conjugate side. Now, simultaneously, we also did for the small molecules, say using fatty acid conjugates for temozolomide, where we have done conjugation with oleic acid, palmitic acid, and linoleic acid. So these different acids, fatty acids were used. Again, these fatty acids were converted to their hydrazide derivatives and then reacted with the temozolomide carboxylic acid derivative to obtain the conjugate. So we have this temozolomide carboxylic acid derivative. Say in this case, the data shown over here is for the oleic acid. Oleic acid is converted to its hydrazine derivative and then reacted with the temozolomide to obtain this temozolomide oleic acid conjugate. So these conjugates were again thoroughly characterized for their using 1H NMR, carbon 13 NMR, the mass analysis using HRMS, where the data clearly showed that the conjugates were successfully synthesized. Now, next is on the assessment, that can these conjugates also were efficacious when given to the cell culture models. So basically, we selected two different cell lines. One is the C6 glioma cell line, and another one is the U87MG cell line, where in both the cases, the conjugates have shown appreciable cytotoxic efficacy. There is an improvement in the activity of all the conjugates, the three conjugates that we have prepared. So they were compared with the free temozolomide, free acids, and their physical mixtures. Following which, the next step is the mechanism, that how it is happening and whether they were also good enough to suppress the resistance is one of the case which is happening because of MGMT. So we also went ahead to check the MGMT levels in these cell lines. So over here you can see the data that in case of control, the MGMT levels, and when TMZ was treated, so there is an upregulation. The amount of MGMT is increased, that is indicative that these cells are growing towards the resistance side. But when these were treated with these conjugates, there was no increase in the MGMT levels, indicating that the resistance could be overcome in case of these conjugates. Plus, these conjugates have shown a marked apoptosis in these cell lines, both early apoptosis and late apoptosis both have been increased. So similar results were obtained in another cell line also, U87Mg cell line, where again we have seen the expression for the MGMT and then the apoptosis for both, for all the conjugates that we have synthesized. Moving ahead for the in vivo activity, that how these conjugates, whether they were active in the in vivo conditions when they were given to the animals. So for that, we have developed orthotopic glioma model using C6 cell lines. So what is done in this case, that the C6 cell lines, they were injected in the frontal cortex. They were injected for the animals, indicating that the model has been developed. So now the next part is testing these conjugates. So among these, we have taken the oleic acid conjugate of temozolomide to test that, wherein the therapy was the injection is done on the right side, and we can see that in the positive control there is a increase in the ratio, while there is a decrease when it was treated with these temozolomide as well as conjugate, and also the brain weights were taken. So survival analysis was also done, wherein it was shown that in case of positive control, all the animal dies within 20 days. While there is an improvement, definitely temozolomide is a first line treatment, so there is an improvement in the survival. And in case of the conjugate treated, we didn't observe any animal death till 80 days. So again, so these are basically the data that we have obtained for analysis, and the studies are again ongoing, where we have further taken these for the analysis, immunohistochemistry as well as histopathology analysis for these tissues. So that those studies are still ongoing, and also the studies for the polymeric conjugates of temozolomide, they are still ongoing. Now this is all with regard to that. I also take this opportunity to highlight the firm that we have created, which is Nanobreed Innovations Private Limited, that is basically working towards the commercialization activities of the nano-based products, because we know that these nano-based products, there are challenges which need to be addressed, and we need to bring them for the commercialization activity. So this company is basically working in that direction. Now finally, the acknowledgements. No research is complete without the financial support, as well as the support from the students who actually spend day in day out working in the lab tirelessly. So basically I would like to acknowledge the financial support that has been obtained from ICMR and DVT for conducting the research, and also the students, Reena, who basically worked for the small molecule conjugates, and Prabjit, who worked on the polymeric conjugates, and Deepak Sahel, who worked for the animal experimentation on developing these models and testing these formulations, and also the collaborators who contributed a lot at various aspects, right, starting from the sciences, like Professor Sakuja, who is a chemist, who assisted us in the design and the synthesis part. Then Professor Sujata Mohanty and Dr. Manu Talela from AIMS, who helped us in various bioimaging experiments and animal work at AIMS, and Professor Anupma basically in the cell culture activities. So she has assisted and helped us a lot in that. So with this, I would like to thank the organizers and the audience, and I'm open for any questions.
No one's
Charan,
asking. I can.
Yeah. Yes, Dr. Bishop.
Very nice talk, Deepak. And very interesting work. I have a few questions I might have missed. For this glioma model, So were these animals, the dosage, was it oral gavaged or injected?
So this was injected in these animals. So
Okay.
initially we started, it's intravenously we have given these molecules. But our
Okay.
next plan is to go with the oral dosing as well. Because initially we
Okay.
want to see that these were efficacious, because we right now we don't know the oral bioavailability. Those studies are also ongoing, but first
Yeah.
is to assess whether they are going to the tumor tissue and showing the efficacy. So we went ahead
Yeah.
with the intravenous initially.
Yeah, very interesting. And the second question I had was, like, I see you are covalently conjugating the drug to your polymeric or small molecule systems. When they are covalently conjugated, so how are they coming off? That's one question. And once you conjugate, FDA or any regulatory body would consider it as a new moiety.
Yes.
So how do you think, would it be easy or challenging to get, approvals, once the testing and everything is done?
Yeah. So basically, coming to your first question about the conjugation side. So if you look at the mechanism, right? I'll just go to that side, because it is basically the methyl diazoniamine which we need. So that can come from that conjugate itself. So it's not necessarily that the bond has to be broken down, because we are having this NH-NH linkage. So this temozolomide, basically it is converted to the AIC and then to the MTIC and then to the AIC, giving the diazoniamine, and it is that methyl diazoniamine that is showing the activity. So that is the first part. And the another part is on the NCE side. The FDA definitely, since it is a covalent linkage, it is considered as a new chemical entity, and there are challenges whenever we are designing these systems. It has to cross all the clinical trials for the safety and then to the efficacy side to get into the market. That is a challenge with any polymeric conjugate system or a small molecule conjugate system.
Yeah, I agree.
But what happens with this molecule, that if you go with physical loading, the challenge comes on the loading of this molecule and then the release side. So there have been reports where say loading of around say 5% is obtained, but whole of the drug is coming out in say initially 6 hours.
Yeah.
How much good it can do? That is always a questionable thing.
Yeah. And for the oral uptake, the studies you're doing, you will, I'm assuming you'll do Kakutu monolayer work and something like that, or you're directly proceeding into.
Yeah, we have some initial data for the oral bioavailability of this molecule. We initiated, in the animals basically, not in the Kakutu cell line.
Okay.
Plus the idea for having these fatty acids, because they can also enhance. There have been reports that they can enhance the permeability across the blood-brain barrier as well. So that was another idea for putting up. So our initial results, although I have not that data right now handy with me, but initial results, what we have observed, that the permeability to the brain is increased for these fatty acid conjugates.
Yeah. Thank you, Ibro.
So I think, Charan, if there are no more questions, you can just take over.
You are muted, Jared.
Wonderful talk on the topic of fatty acid conjugates in the drug delivery system technology by taking an example of temozolomide, which is an anti-cancer agent. So this is good to see the entire data. So for the synthesis of the conjugate, the role of fatty acids in the same chemical reactions is concerned. So you have shown a lot of data in terms of the synthesis followed by the characterization of the sophisticated techniques like NMR, HRMS, and others. And thereafter, you spoke about the evaluation of the same in terms of cell line studies, and the C6 cell lines and UAT7MD. These are the two well-known cell lines for the study of tumor cells. And lastly, sir, it was nice to see the data in terms of the orthotopic model. And so far as I know, this is a very difficult model to use in the lab by injecting the cells. And beautifully, you have shown the histology of the brain also. And on the left and the model. So overall, it was an informative talk, sir. And as a token of love on the behalf of ISFCP Pharmacy, we like to present a certificate of appreciation. Yeah, I'm just about to share the same.
नहीं ऐसी डॉक्टर चरण जी हेलो
जी सर
Professor YK Gupta sir is here, so he want to also interact a few minutes with the speaker. And
right sir,
just three speaker is here: Dr. Deepak, Dr. Tushar, Dr. Banta Kakkar. Yes, you all are pharmacists.
So sir, I invite you all, especially
Good
Professor
morning,
YK
sir. Very
in
good
the,
morning.
Good morning. Good morning. Vandita, how are you?
I'm fine, sir. How are you doing?
Fine. Thank you. No,
So good to see you.
pleasure. अभी इनका लेक्चर शुरू नहीं हो रहा है उधर कहीं लेट नहीं है एंड हुआ है अच्छा एंड हुआ है अभी तो शुरू है
एक लेक्चर हो गया है डॉक्टर दीपक जी का
और ऑडियंस वेट तो नहीं कर रही नहीं okay so wonderful meeting you all Deepak and Tushar. I think let us have a quick round of introduction. I am Dr. YK Gupta here sitting in front of you and I feel pleasure in interacting you. So Deepak, where are you now?
Sir, I'm in BITS Pilani in Department of Pharmacy in Rajasthan.
Very good. And where you did your PhD and your M.Pharm?
So my MS Pharm in pharmaceutics is from NAAC Mohali, and then my PhD is also from NAAC Mohali. After that, I went in an exchange program at University of Tennessee Health Science Center for around 1 year, following which I did my postdoctoral research at University of Nebraska Medical Center, and then finally 8 years back I joined at BITS Pilani.
And now you are as professor there or as an associate professor?
Associate professor, sir.
Very good, wonderful. Nice meeting you, Tushar. आप कहाँ पे हैं अभी?
Hello, Dr. Gupta. I am in Sydney, New South Wales, Australia. So I am a senior lecturer here in material science, and we work on biomaterials. I did my bachelor's and master's in India, and then I came to Australia to do my PhD. And
sorry, your voice is very low. You did your master's from where?
I did my bachelor's and part of my master's from India, and then part of my master's from University of Adelaide.
In India, which institute in India you did?
Amity.
Sorry?
Amity in New Delhi.
Amity?
Yeah, I did my bachelor's and part of my master's from Amity, and then PhD from University of Adelaide, and part of my master's from University of Adelaide. And then I did a postdoc at UC San Diego, University of California, San Diego, and then I've been at University of Queensland, and then now I'm a senior lecturer at University of New South Wales in Sydney. So I've traveled around as well, like Deepak.
Wonderful. And Vandita, what about you?
Sir, I am a product of UIPS Punjab University, where you have been coming. So I have done my bachelor, my post-graduation, my doctorate, all from UIPS Punjab University, and I've been working with pharmaceutical company like Fresenius Kabi and Venus Remedies. After that, I joined here in 2012. And
where are you now?
University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh.
Oh, no, you're back to Punjab UIP.
Yeah.
Oh, very, very good. So it's pleasure meeting you, Deepak, Tushar, and Vandita.
Thank you, sir. Same here.
Pleasure. Namaste.
I've heard a lot about you, sir.
Yeah, we were fortunate to have sir as a mentor, as a supervisor for the DBT project, which Professor Indu Palkor was having. So we had sir here for the due diligence. So I hope you remember.
Oh, yes, yes, yes. You were there at that time.
Yeah, I was there.
Okay, great. So I think we will, maybe we can have an opportunity to meet. I do not know with Tushar or not, but at least with Deepak and Vandita, we will have an opportunity to meet
Definitely.
in person sometime. But Tushar, whenever you come
Yeah.
to India, I think let's have let's meet sometime.
Most
And
definitely. I think I'll be there in August.
this meeting which
Yeah.
is being organized as a Ibro workshop is a very important meeting. I would say. Why it is important? Because the graph of neuroscience research in India is declining fast. Say when we were doing neuropharmacology, it was at low, but then many scientists started working, including my friend Shyam Sundar Sharma in Naipur and in Punjab University, and so many places people started working on model of stroke, including CDRI, IITR, and many places. So there was a large group of neuroscientists, but soon the scientist, I would say, cadre, true scientist cadre, is declining, and that too, the more casualty has happened in neuroscience, and therefore your leadership or your mentorship to the new generation is very important. And what I would say, when you talk, when you give a talk, or when you tell, don't tell what exactly you are doing. Don't glorify your research. This is not the forum for that. Usually we do that when you are giving some oration or some lectures. But this is the time when I would suggest that you convert your talk in a mode of training, teaching principle of neurosciences, how they should do it. Don't make a robot. Try to make your students or your audience a thinker. That yes, if they, because neuroscience, particularly if you talk of a stroke, you talk of Alzheimer's, epilepsy, none of the model is what you can say is a perfect. But and therefore you have to use a more appropriate, I'm not saying most appropriate, more appropriate model, and perhaps you can also tell them what is the need of having multiple models, and then you must also tell them what are the caveats. In fact, I listened to one talk yesterday, and what are the caveats when you do this research, maybe stroke or Alzheimer's or epilepsy, or maybe in isolated tissue. That caveat is that why this is not can be considered as 100% possibility in human. What could be the caveat here and there? And that is why when you say interplay of neurons or interplay of neurotransmitters, it is influenced by so many factors, and these factors should be taken into consideration by all the scientists. The second thing, being chairman of National Technical Committee of GLP, I have started a program from DST to train the academic institutions in principles of GLP, good laboratory practices. That is, they should be told how to keep the record of the data with them, how to preserve them, because the integrity of the data is so crucial. Because every time we cannot say this is not reproducible because they have used different methods, they are using this and this, but the data must be reproducible. That is the spirit which you as an ambassador should try to convey to them. Keeping register, keeping the animals in good condition, ethical. So these are something which are very important, and people are missing these things. And I have seen people sweating when you ask raw data from them. So these are the things which all the audience and all the students should be reflected upon, because Indian science has now recovered from a very depressed stage. Now Indian science is respected. We are publishing very good, still much lower as compared to our scientists versus the articles published. But more important thing, how your research is respected. Impact factor is less important than the reproducibility factor. Remember this. I'm not bothered about the impact factor. Your entire impact factor, the reputation of the institute is crushed if your data is not reproducible by doing so, or the data is questioned. So emphasize this again and again wherever you get a possibility. And lastly, I would say it is important to collaborate, because in isolation, work in isolation, gone are the days. You have to collaborate, inter-laboratory collaboration, inter-institution collaboration. Caveat, the most important thing is never ever give a authorship which is a gift authorship. Never. Or which is a reciprocal obligation authorship. I've seen people making author of international university without their any contribution. Ridiculous, unethical, illegal sometimes. Because, and never ever accept authorship by yourself, even if you have contributed some degree of intellectual thing in that. That can come as the acknowledgement. Why I'm saying so? Because I come across a couple of committees which are inquiry committees for setting up these things. So because you are not only taking lectures here, but I would say you are an ambassador to young generation scientists. So I look forward for interacting with you, Vandita, Tushar, and Deepak, and I can see Parneet Kumar also, and of course I can see Dr. Charan. So wonderful. So बहुत अच्छा लगा मिलके, and maybe we can have some time. Great.
Thank you
Thank you, sir. Thank you so much. I'm looking forward to meet you very soon.
Yes, yes, Vandita.
So, thank you so much, sir, Professor Mike Guptani, for having your utmost important input so far, the status of the Indian science is concerned. Sir, each and every point of view is important for us. As you rightly said, Deepak sir, Dr. Tushar, and Dr. Vandita, they are ambassador to the neuroscience research. So thank you so much for sparing some time with us in this particular meeting, sir. So as a token of love, on the behalf of myself for the Pharmacy Moga, we are going to present the certificate of appreciation to Dr. Deepak. So I'm just sharing the same. It's, please give me a few seconds.
वंदिता, तुमने ये अपनी फोटो कब की लगाई हुई है इस पे?
सर ये हो गई 2 years back.
Yes.
Good, good.
Around 2019 की है.
कर लें एक साथ हम लोग फेलिसिटेशन कर लेंगे.
हाँ जी.
नहीं, आई एम टॉकिंग
चरण जी तुम लोगों को माला पहनाना चाह रहे हैं, लेकिन वो
सेकंड लेक्चर चालू कर लें so that we can felicitate at a time all speakers.
Sorry, sir, for this technical glitch. Deepak sir, actually, this is not appearing. I have these certificates, but due to some technical issues, I'm able to.
That's okay, sir. No issues. That's fine. Yeah.
Thank you, sir, for your wonderful talk.
Thanks, Deepak. It was very nice.
So, sir, we move forward to the second lecture of the technical session with Dr. Tushar Kumaria from UNSW, University of Science, the University of South Wales, New South Wales, Australia. So, here is the I think it's again not visible. The introductory slide.
Charan, I can introduce myself. Don't worry. If there is an issue, I can do a brief introduction.
So, for the next lecture in the technical session, belongs to Tushar Kumaria from UNSW, Australia. So, so far the introduction is concerned of Dr. Tushar Kumaria. So, as we had a chance to have him in the previous technical sessions of the ISFCP Pharmacy Model in a series of lectures. So, so for today's lecture is concerned, he is going to speak on the porous materials in drug delivery and imaging. And he is a senior lecturer at the School of Materials Science and Engineering at UNSW Australia. He has received his PhD in 2015 from the University of Adelaide with a doctoral thesis medal and Dean's commendation letter. He has spent the following 2 years at the University of California, San Diego, UCSD, USA, as a postdoc professor Sailor's Lab, and returning to the Australia in 2017 to a prestigious early career fellowship at the University of Queensland, Australia. He has co-authored over 90 peer-reviewed publications in top-tier journals in the field of nanomaterials, biomaterials, drug delivery, and consequently have attracted more than 2,800 citations on Scopus and returns an H index of 33 and more than 3,300 sites on Scholar with H index of 36. Additionally, he has successfully secured over $4 million, and out of $4 million, more than $2 million as a lead CI in competitive research grants, including an NHMRC Early Career Fellowship, ARC Discovery Project, and Rama Quality Health Investment Grant, which is a US Department of Defense grant. His research focus is on the development of smart porous materials, or we can say mesoporous materials, and their composites for drug delivery, bioimaging, tissue engineering, and diagnostic applications. So, Tushar, we are happy to have you in the technical session of the day 2 of Ibro Spotted meeting and workshop. So, this is over here for the technical session.
Thanks, Charan. I'll start sharing my screen. It says you must grant permission. So can you see my screen now? Charan, can you see my screen?
Yes, yes, it's visible.
right, shall I start? Okay, so I believe everybody can see my screen. I'll start sharing. Thanks, Sharon, for this nice introduction. Can you mute yourself, please?
Yes, it is.
Sharon, can you mute yourself, please? Thank you. Right, so I am Tushar from University of New South Wales in Sydney. I am a senior lecturer here, as Sharon said, and my lab works on a range of different types of porous materials for drug delivery and biomedical application. We have a very small focus on nervous system regeneration field and nose to brain drug delivery application. I'll give you some examples of those as well today, but a lot of focus on oral delivery of payloads. As Deepak mentioned as well, you know, there's plenty of drugs which have a huge challenge for oral delivery. They either degrade, they don't permeate, their solubility is low. So this is what we work towards, solving these challenges for a range of different type of drug molecules. So I'll show you a few. So before we start, I won't cover, you can see these colorful images. These are porous silicon-based photonic crystals. I won't talk anything about this. Mostly what I will talk about in my talk today is porous silicon and mesoporous silicon dioxide-based materials. We do work on titanium nanotube materials for localized delivery application in implant and implant surfaces, but I have not covered it. We've done porous alumina work for sensing applications. So the whole focus of the lab is on porous materials, but for drug delivery, we mainly use porous silicon and mesoporous silicon dioxide. That is what I will share with you today. I have few things that are not published, so please request if it's being streamed online.
Madam.
So there's Prateek. Can you please mute yourself? Okay, so I have a few slides which are unpublished. So can I please request that they be paused from sharing online if it's being uploaded to YouTube? I've put a note on it, so it should be easy to spot. All right, so in terms of my journey, as I already mentioned, I am from Delhi originally. I did my bachelor's and master's, a part of my master's from India, from Amity University. After that, I moved to University of Adelaide to do my PhD and part of my master's degree. And once I finished my master's degree, I joined the same group to do PhD on the same topic of porous materials. And once I finished my PhD, I moved to University of California, San Diego, to do a postdoc. I stayed there for 2 years and mainly focused on porous silicon-based materials for a range of drug delivery applications. I'll show you a few. One of those was on nervous system regeneration using composites of porous silicon material and their nanofiber composites for, this thing, what do you call it, spinal cord injuries. In 2017, I received a fellowship that allowed me to come back to Australia. So I moved back. I stayed at University of Queensland in Brisbane for almost 3 years. And in 2020, I received 2 major grants, almost $1 million in total, that allowed me to move into the current position I have at University of New South Wales, which is in Sydney. And it's one of the top universities. Our school ranks #2 in Australia and number 36 in the world in QS World Ranking 2021, 2022. And so we do a range of different things. And I am a materials scientist by training with the major focus on making biomaterials here. All right, let's start with what we do in the lab. And Deepak also mentioned about safety of the materials that we need to design before we consider anything else, right? And this is where my talk starts as well. So I have this image here. This is a book, I, Robot, from Isaac Asimov, which gave us 3 basic rules of robotics, right? And these 3 basic rules of robotics allowed us to design, computer control systems and follow these 3 basic rules for safety from these, robots and computer design systems. So in terms of robotics, a robot not injure a human from its, from its action or inaction. That's the rule number one. Rule number two is it must obey human unless it conflicts with the rule number first. And it must protect its own existence unless it conflicts with rule number 1 and 2, right? So these are three basic rules that were given to us by Isaac Asimov. I think we need similar rules when designing nanoformulations, right? And the reason we need these is so that we can make the nanomedicine safe and efficacious, right? So three rules that we follow in our lab while designing systems is, the first rule is that the system should perform the way it, or the function that is designed to perform. Say for instance, if the system is designed to do drug delivery, it should do that efficiently. If it is designed to do bioimaging, it should enable that. If it is designed for both, it should perform that function quite well, with high efficacy. Second rule is that it should be able to target the disease site. If it's IV injectable, if it's orally injectable for specific disease things, say inflammation, it should be able to target the inflamed site in the GI tract, right? The third thing is that the system should clear out from the body safely without leaving any harmful trace. Most of, organic systems that you see, most of the nanomaterial systems that you see in the market, if you look at this, so I don't know why there's a delay. It moved 4 slides instead of 1. So what I was saying is the system should clear out from the body without leaving any harmful traces, right? And most of the organic systems perform this function quite well. And you can see this in the nanoformulations which are either approved or are in clinical trials, and if you look at this list, most of these formulations are either polymer-based or lipid-based. The only few inorganic materials-based formulations that you see here are iron oxide nanoparticles, which are less than 10 nanometers or so they get renally cleared. That is the only inorganic nanomaterials that you see in the system. This is Professor Sameer Mitra Gothri from Harvard. He was at University of Queensland in 2019, and I took this photograph. I think it summarizes the field quite well. If you go through all of these, there's only a few inorganic materials. And the only reason is most of these, they don't clear out from the body efficiently. So the focus of my lab is to design materials which are inorganic porous materials, but perform all three functions efficiently, right? Or if they don't degrade, we don't use them for other specific application. I'll discuss those in detail. The main method of making the materials in our lab is common. So we utilize this technique called electrochemical anodization, in which we have a working electrode, which is either silicon or aluminum or titanium, and then we drill holes into them using electrochemical etching, right? So what happens in this process? You have an electrolyte, you have a counter electrode. The working electrode, as I mentioned, is the material. And then when you apply current, it drills holes in the surface. And I've got a video to show you for that. So in case of making porous silicon, we start with silicon wafer, as I mentioned. We pack it in this etch cell, apply current. And what this leads to is, it leads to formation of this porous layer. If you remember the first slide had these colorful chips. These colorful chips are basically the same photonic crystals, porous layers, which are still attached to the silicon wafer. Whereas for drug delivery application, we can lift this off from the surface of the silicon wafer, and what we get is freestanding films, which we break into micron or nano-sized particles for drug delivery, right? So you'll see some of these porous silicon particles here. These are flat sheet-like particles. Mesoporous silicon dioxide that we make in the lab is from Sol-Gel silica process. I've not covered it. It's quite common. You can find it. You find the whole reaction very easily. If anybody has questions, you can reach out. I am very happy to give more information. But this is what our particles look like. And the reason we use these for drug delivery applications is, one, we can tune the pore size. And what that allows us to do is we can change the type of payload or tune the pore size of the material to the requirements of the payload. Similarly, we can tune the surface chemistry. We can have particles which are hydrophilic or hydrophobic. And then by tuning that, we can tune it to the requirements of the drug molecule, to hydrophilic or hydrophobic. And we have tested these materials in a range of different cell lines. Other people have as well. So we know that these are cytocompatible. Being porous, we achieve very high drug loading capacity. So most of the material is actually air. So there's very large open pore volume that can be filled with drug molecules. So in case of proteins, we are able to achieve drug loading capacity in excess of 48%, which is amazing and not achieved by any other type of liposomal material or polymer-based materials. We can tune the drug release based on particle size, particle surface chemistry, and the pore size. And the main thing is that these porous silicon materials, they degrade out in the body into a non-toxic silicic acid form that gets cleared out. And silicic acid is the most bioavailable form of silicon in your body. So it's non-toxic and gets cleared out. The other advantage is that we don't need to tag our material with any fluorophore, because our material by itself shows intrinsic photoluminescence that we can use for tracking where the particles are. And I'll show you some of the examples where we use this for bioimaging. So what we do with porous silicon materials in our lab is we use them for a range of different drug delivery, systemic, oral, and local delivery for proteins, small molecules, and genes. We do tissue engineering with them. We use them for bioimaging and sensing. Most of what I will cover today is protein delivery, small molecule delivery. A few examples of tissue engineering with these and their composites, and some examples of bioimaging. All right, so let's start with drug delivery in terms of like where we utilize the specific photoluminescence properties of these porous silicon particles for drug delivery into the eye. This formulation is targeted towards age-related macular degeneration. So people who have age-related macular degeneration, they need to go into clinic and get an injection jabbed in their eye every 4 to 6 weeks. So there's frequent injections. And the second thing is the doctor relies on reoccurrence of disease symptoms to decide when is the next dose time. So what, the two requirements for the field is, one, can we extend the time period of the dose? And two, is it possible to track how much drug remains in the formulation itself? So this is what these formulations, photoluminescent porous silicon formulations, allow us to do. So if you see these particles are photoluminescent, and when they degrade, the photoluminescence goes down. And as the photoluminescence goes down, the drug payload that is loaded within the pores gets released. So we can track how much payload is released to how much the decay in photoluminescence is. So we can get a formulation which is either a zero-order tracking system or a real-time tracking system, or a system which acts as an end-of-life indicator, right? So in this case, depending on the thickness of the oxide shell, we can either get a system which tells us at each time point how much drug is released, or a system which does not show any decay in photoluminescence until 80% of the drug formulation goes out. So this system acts as an end-of-life indicator. And doctors, this is a very good system for clinical application. Why? Because doctors would know once the photoluminescence drops below a threshold, they know that more than 80% of the drug has released out, so it's next time to do the injection. We move these particles towards in vivo applications, and we've done first a few safety trials in rabbit eye, and we've looked at the photoluminescence decay of these particles in rabbit eye. So what you see here is these porous silicon particles injected into rabbit eye. These brown, boluses are porous silicon particles. This is a color image, and this is a bright field image with a blue filter on it. And what you see here is this is a without the blue filter, and this is with the blue filter. So what you see is the photoluminescence of the particles themselves. So by stitching a few of these images together, we could track the decay in photoluminescence of these particles. And if you look, the in vitro photoluminescence decay and in vivo photoluminescence decay track quite well together. So that's quite encouraging for us. We are looking to do, we've taken eye taps from these rabbits. So we plan to do analysis of these eye taps to track how much drug gets released versus the time photoluminescence decay in vivo as well. But that we believe will take some time to finalize. All right, so moving on, as I mentioned, we do drug delivery in the lab, right? So one of the focus in the lab is oral delivery, and oral delivery specifically for a range of different type of molecules, either with solubility problem or permeation problem. So this formulation here is targeted to.
Sorry to interrupt you. Would you please reduce this size of the slide, because the title are not visible. These are half cut down.
Sure.
The title of
I can see it complete. I don't know why it's not appearing.
and, slides number. I'm more worried about the slide numbers, as you said.
Can anything change? Right, is it better now? I can like this.
I think this is 10th, 14th slide, right?
This is 10.
10. This is 10th slide. Okay. I think slide now. The title.
Can you see it better? I will tell you which slide number to stop. I have put labels on it, so there's no
Problems. This is fine.
Okay, perfect. So as I was saying, meropenem is a late stage antibiotic, and the issue with the drug is very poor permeation, and this poor permeation is because of PGP efflux pump. So if you take meropenem, it gets through the lumen, gut lumen, but gets spit back out into the lumen because the PGP efflux pump spit it out. So the idea was, can we create formulations which act as a biomodulator, which can block or act as an antagonist for the PGP efflux pumps? So we functionalized porous silicon particles, and the other reason was to increase the stability of the drug. There's very poor stability. This drug degrades really, really quickly. So we functionalized our porous silicon particles loaded with the drug, and then we looked at how well does it provide us. It's really slow. So after that, we looked at how well can we control the permeability of the drugs. So we have a CACO2 monolayer model in the lab. We looked at secretive and absorptive, drug absorption or drug permeation. And if you look at the efflux ratio is how much drug goes through secretive pathway versus the ratio to how much of the drug goes through absorptive pathway. And meropenem has a very high efflux ratio. It means a lot of drug that is fed into basal chamber comes up into apical chamber. Whereas when these functionalized particles are added, we can reduce this efflux ratio so that more of the drug retains in the basal chamber, which represents the blood circulation. So that's an advantage of using these particles and making particles which are biomodulating. This can be used for modulating drug permeation in case of many other drugs as well, like what Deepak was talking about. PGP efflux pumps are an issue for a lot of brain tumor drugs as well, temozolomide itself as well. So that's, this is why we are investigating these drug molecules. Most of the focus of the group is on oral delivery before moving into cancer studies or brain targeting, we want to functionalize and figure out the systems for oral delivery. Once they work, then we move on to looking at glioblastoma and other type of brain tumor models. This just shows the activity of the drug, that the drug is still active. The issue with this formulation was that the drug releases out really, really quick. There's no control over the drug release. So we moved on and we designed a system where we had a coating of urigid-based polymers, and we looked at if these urigid-based polymers can, reduce the rate of release of the drug or provide some control. So we could see that, after coating the drug-loaded particles with these different type of urigid samples or polymers, we can get a more controlled and better release profile, which extends to 8 hours. So it means that drug is stable for up to 8 hours, whereas drug by itself degrades out really, really quickly. Within about 2 hours, more than 40% of drug degrades out. Whereas in particles, we believe the drug remains active. We've done LCMS to quantify and figure that out. So this presents, the amount of drug degraded and the amount of drug that is active. So this curve shows the ratio of the drug that is degraded over time. So if you look at cumulative, it's quite low. Again, we wanted to look at do these particles by themselves act as an antagonist for reflux pump or not? The other type of functionalizations that we explored, they do, but there's no control over drug release. Whereas the urigid systems do provide control over drug release, but we want to figure out if we can still control the efflux pumps or not. So what we found that one of the urigid polymers, RSPO, works quite well. It reduces the efflux ratio significantly compared to the drug itself. So herein, we confirm that we can enable controlled release of drug, as well as we can control the efflux pumps or biomodulate the interaction of cells just by modulating the surface functionality on the particles. This is some of our unpublished work. This is targeted for nose-to-brain delivery of drugs, and mainly small molecule drugs which are hydrophobic. The challenge with hydrophobic drugs is that one is they tend to crystallize out in liquid-based systems. Second is their solubility. They don't get uptaken because they are crystalline. So the idea was, can we load these drugs into porous silicon carriers and then embed them into hydrogels? So what these hydrogels do is, when they are sprayed into nose, they adhere to the nasal cavity and gel out. And once they gel out, they stay there for longer time, providing more sustained and controlled release in the nasal cavity. And what that provides is it provides a more efficient nose-to-brain delivery. It also prevents leakage of the drug into the back of your throat, right? Because if you spray any liquid formulation, you would know if you have ever taken medication for nasal inflammation or cold. If you spray things in your nose, a lot of formulation goes towards the back of your throat. So this prevents that. So this is just showing, when we add different drug amounts, this is a 0.18% drug. The drug we chose was mometazone furoate. It's a very lipophilic drug. This formulation, 0.18% equivalent, is already present in market, but we want to try if we can increase the amount of drug that we can incorporate in the formulation without disturbing the gelation time a lot. So the hydrogel by itself has a gelation temperature of 27.4, whereas 0.58% equivalent drug formulation has a temperature of 28.5. So this shows the release profile. What you see here is the cumulative amount of drug released from the hydrogel itself, from 0.18% equivalent drug, is only 0.4% after 72 hours. Whereas the formulations which had porous silicon loaded drug particles, porosilicon particles loaded with drug, we get about 8 to 10% of the drug release out within the same time period. And as we increase the amount of drug equivalent in the system, we see more amount of drug being released out, right? So this is quite interesting. And we believe that this is because of when we embed the drug inside these pores, the pores are too small for drug to crystallize, and drug remains as a polyamorphous solid, and it's more bioavailable and more soluble, and that's why it gets absorbed better. We did toxicity studies of these, drug formulations on human nasal explants. So we took human nasal explants from a hospital that we work with. The only formulation that showed toxicity on the human nasal explant was the positive control zinc sulfide. This is the most interesting set of data that we have. So what this shows is at 8 hours time point, when you load the drug by itself into hydrogel, in the human nasal explant, only about 10 to 20 micrograms of drug gets absorbed per gram of sample or your tissue weight. Whereas when the formulation is based on porosilicon hydrogel, we get about 200 micrograms per gram, which is 20 or close to 20 times higher than the drug by itself. So this is very exciting. So we are moving ahead with this, and we are trying more experiments with this. We'll aim for, we'll try other type of drugs as well for nose to brain. And we will also look at protein-based formulation if we can achieve protein permeation or protein-based drug absorption as well or not. All right, so this is a work on nervous system regeneration or tissue regeneration. So what you see here is we devised a new type of nanofiber fabrication or nanofiber scaffold fabrication technique using our porosilicon particles loaded with protein-based drugs and polymers as the backbone. So what this technique allows us to do is to make these nanofibers directly onto any type of surface. And we can orient these fibers into any directional alignment as the requirement needs, right? So when we look at nervous system regeneration, one of the requirements is that the alignment of neurites has to be guided along, right? If somebody has a spinal cord injury, the neurons need to be guided so they can make linkage again. Whereas if that does not happen, neurites grow everywhere, so the linkages are lost. So this is what the intended purpose for making these directionally aligned nanofibers was. So that's what you see here, these porosilicon particles loaded with protein payload embedded in these nanofibers that we made. So these are randomly oriented fibers, and these are directionally oriented fibers. And the model protein we looked at or we chose was lysozyme. So when the lysozyme is directly added into particles by itself, and over 200 hours, we achieve almost 100% bioactivity retention from the lysozyme that gets released. And when these particles loaded with lysozyme are embedded in these nanofibers, we achieve around 70% activity retention. This is quite substantial given the loading capacity of these particles for lysozyme and other protein-based systems that we have tested is in excess of around 60 to 70%, 30 to 40% loading capacity. So, a 1 milligram formulation contains about 30% of the drug. So these are the, some confocal microscopy images where we have shown that when these neurites, these are dorsal root ganglion cells from rats. So when they are grown on a flat substrate, you see these neurites growing everywhere. However, when these are grown on the nanofibers, the composite nanofibers that we have, we can see directional alignments of these neurites from the central body. And this works well for large cells like DRGs. So we wanted to look at, can we achieve the same thing for smaller cells like Neuro2A? And that is what we've seen. We can see more directional alignment, and you can see this from this photo plot. The other, as I mentioned initially, that this technique allows us to make nanofibers on any kind of surface. So one thing we want to, or we are very interested in looking at is, can we make these nanofibers directly at the injured tissue or not? So that's one of my goals. So we went around and we looked at other type of polymers. We looked at, we optimized the spraying condition based on a lot of different parameters. So we looked at working distance and time. We looked at sample collection platform rotation. We looked at sample collection placement, collector placement. So if it is placed, directly under the nozzle or offsetted from the nozzle. And we looked at nebulization time. So can we control the density of the nanofibers per centimeter square or not? So when we optimized, we were looking at, we were also looking at the mechanical properties, because for a nervous system regeneration scaffold, mechanical properties are also important. So based on, so what we figured out was that the rotation of this platform helps in getting fibers which are more homogeneous. The other thing is the system that works best is around 20 to 25 centimeter away from the nozzle. And in terms of sample collector placement, offsetted sample collector placement is better because we get less globules formed into the system. In terms of the mechanical property, this is the major results of what we see is ultimate tensile strength increases slightly, and load at failure increases as the amount of fiber density increases as well. We then looked at how cells grow on these as before, but this is the most interesting set of data. So we looked at what happens, or can we directly spray these fibers out onto live cells or not. These fibers, the backbone polymer is polycaprolactone. It dissolves in dichloromethane or chloroform. Dichloromethane, if you add it to cells, they die, right? But what we see here is by our nebulization-based technique, when DCM by itself is sprayed, the cells remain alive. So this is DCM by itself. And this is the ambient control air. When we treat the cells, we spray for 30 seconds for cells. So we wanted to keep the cells outside for 30 seconds as well. This is ambient air that is blown onto it, and a range of different parameters. These are the nanofibers sprayed onto live cells. So this is live dead staining, right? So the green dots that you see are live cells directly sprayed with nanofibers for 30 seconds using a dichloromethane-based polycaprolactone solution. So it proves that dichloromethane gets evaporated during the spraying process and cells remain alive. Whereas when dichloromethane is added in liquid form to cells, they all die. So, it shows that we can directly spray nanofibers onto live cells. So we are moving ahead, and we will try, more application as to now look at, can we embed protein-based systems that also act as growth factors, or other type of, anabolic growth factors that we can add to not just directly spray the fibers onto the cells, but also promote their growth or not. How much time do I have, Jared? I'll keep on going for another 5 minutes or so, and then I stop. This is a bioimaging work that we've done recently. I'll skip this, but mostly what I want to show you is we have done a lot of photoacoustic imaging, stabilizing an FDA-approved dye that is used for both fluorescence and photoacoustic imaging. I'll not go into details of this, but I will go into details of this. So this is unpublished data. If you can please not post, that would be great. But basically, photoacoustic imaging, what that is in case of ultrasound, when you, if somebody goes for ultrasound imaging, what tends to happen is you shine ultrasound, you pick up reflected ultrasound waves. In case of photoacoustic, you excite ultrasound vibration through a pulsed laser. And there are specific materials which, when they are excited, which absorb in near IR region, can, enable that. So in the previous work, we used indocyanine green as the modality, but herein we utilized our own porous silicon particles themselves as the photoacoustic excitation modality. So what this shows is that we were able to make these particles, which are photoacoustically active, and the particle size remains quite controlled even after functionalization with this 10D7 antibody. This antibody is the one that targets specific tumor. This shows the photoacoustic imaging data that at around 700 nanometer excitation, we get the highest photoacoustic intensity signal. And the intensity of photoacoustic intensity of our particle is much more stable over time compared to the ICG itself at the same concentrations. In addition, the photoacoustic intensity is much greater from our particles at same concentration, about 6.2 times higher compared to ICG. Silicon dioxide nanoparticles are our control, which are the starting precursor. It shows that they show no photoacoustic intensity. This is just these particles injected subcutaneously. We moved on to doing in vivo studies after looking at in vitro targeting capabilities of these particles functionalized with 10D7. So what we see is the particles which are targeted, we see more of these accumulate into tumor. This is 48 hours post-injection, and these are controls which are 48 hours post-injection. These are particles which are not functionalized with 10D7. So what we see is initially there's no difference between targeted and non-targeted particles, but non-targeted particles get away or get leached out from the tumor quite quickly, whereas the particles which are functionalized tend to sustain. And this is a significant difference. Please note that the animal number is n equals to 4 and 5, but there are 2 tumors on each mice. So the actual number of images that we've taken is 8 for the control and 10 for, or actual number of tumors that we've imaged is 4 and 10. As I mentioned initially, most of the materials that we work for targeted for injectable applications, they are biodegradable, and that is what you see here. So these particles, they degrade out within 4 hours, about 40 to 50%. Please note that these are unfunctionalized particles. There's no surface functionality, so they degrade out much faster. But in this case, the particles that we've injected are functionalized with PEG, and then on top of PEG, there's this antibody. So we are doing now degradation studies of 10D7 functionalized particles. And right, I'll finish on this. I will not cover this. And before finishing up, I would like to thank all the people who have contributed to this. There's a bunch of people who, without whom all of these work that I've presented would not be possible. Srish did the hydrogel work. Jin Wang and Zainab did the photoacoustic imaging. Even Yukshi and Z did the photoacoustic imaging. Brian is our core facility person. There are key collaborators like John Hooper, who helped us with the animal model here, the tumor model. And these are the institutions which are involved in all of these funding or in all of these research. And currently I am funded by ARC, Australia India Strategic Research Fellowship funding, Visual Sonic, Defense Science Institute, US Department of Defense, and Ramashoti Foundation. So thank you all for listening, and thanks Charan and Dr. G.D. Gupta for this opportunity to speak at this conference. I hope some of our work on nervous system regeneration was quite useful for Ibro people and people working in nervous system or brain diseases. We are doing a lot of stuff for now in brain tumor, or we are venturing into it. So this conference is also an opportunity for me to, find potential collaborators, who might have animal models or who might have, interesting molecules that can be looked at using our particles. So thank you all. Any questions? Charan, you're muted. Charan, you're muted. Charan, you are muted.
Charan, we cannot hear you. You are muted. Charan?
We can't hear you, Charan.
Hello, Dr. Charan. So, any questions from available on the YouTube or any question also expert? So, if you can have any interaction, I think some of the technical problem it may be possible. Dr. Charan, are you hear me?
I just called Charan. He says he's having internet problem. He's not there.
Okay, okay. So within second he will join again.
Yeah.
So, Dr. Tushar, it was indeed a very, very wonderful talk, I must say, and the work is in fact very good. So, are you working on lipidic nanoparticle as well?
We don't work on lipid-based systems, but we do functionalize our particles with lipid for attaining specific targeting. So, for instance, I didn't cover this in detail, but the slide moves. So, the imaging work that I mentioned, so we have tried lipid functionalization of our particles with lipid bilayer to protect the payload inside the pores and control the rate of release. So we've done that before. We have some very interesting work that I can't share right now. It's you know, it's a proprietary project that is funded by USDOD, in which we are designing specific systems which are lipid membrane based, and they are for targeting cytokine storms.
Right. So you're giving them orally, or it is via intranasal route?
So those would be IV.
IV.
We haven't done lipid system for intranasal yet. But if you look at the intranasal system that I mentioned earlier, it's actually based on a tri-block copolymer, pluronic F127, which actually forms micelles and then forms a gel. So it's similar, you know, self-organization is just, it's not lipid, it's a tri-block copolymer.
Right. So while we talk of any commercial viability, how do you foresee these molecules when we talk of, you know, coming to the market for the patient care?
I think they're quite far off. For oral delivery, mesoporous silica and silicon dioxide and silicon-based systems, they are okay. They are already used as excipients. So for oral delivery, there's no problem. And that's why we have a lot of work that I showed is on oral delivery, because, you know, if I pack it into a capsule or a tablet, all good, we eat it already. But for injectable systems, there's no you know, current formulation based on these things. And the main reason is, if you look at all the injectable formulations I've talked about, they are porous silicon. And the reason is because porous silicon degrades, and mesoporous silica, which is silicon dioxide, degrades as well, but at a very slow rate. So that's why we tend to pick particles that, you know, there's a phase 3 clinical trial that has happened with these particles for pancreatic cancer. We don't know the results yet, but safety trials have already happened. Phase 1, phase 2 have already happened.
Okay.
So
they're quite safe. They're very safe.
Yeah. So those have already happened, but efficacy trials for one pancreatic cancer therapy are currently going on, I believe.
Great. It was nice hearing to you.
Thank you, Monica. I am also looking for PhD students. If you have any talented students who are looking for a place, please send them my way.
Definitely. I think we'll connect with each other and we can plan a collaborative project as well.
Most definitely. The other thing I must mention is, I think whoever is here and you know, is interested in collaborative project, I already have in Australia, India strategic funding. It's a very good platform to establish collaboration. So with this, there's Gangaram Hospital in Delhi, which is involved, Amity is involved, On our side, there's 3 universities that are involved, and the amount of money you get is amazing. You can get up to 60 to 70 lakh on Indian side, and on our side, you can get up to $1 million.
Yeah,
And-
Indo-Australia call, I think we can apply.
Yeah,
That's
so
a good option. Yeah.
so I always encourage, and if there's any opportunity, we should look at that.
Definitely we'll do that. We'll do that. We'll take Dr. Charan in loop.
Yeah.
He's also very proactive. Yeah.
Sharon, you muted. We can't hear you.
Yeah, Charan, we cannot hear you.
Sorry, I forgot to unmute. So I'm happy to have in the loop now. We are planning for the industrial static. So surely.
Question.
You are not clearly audible, Charan.
Hello.
Yep. Better now.
Am I audible?
Yeah.
Okay.
Perfect.
So, Pankaj sir and Gupta sir.
Please. Hello. Hello.
Hello. Yes, sir.
Yes, yes. So very.
Hello. सुनाई दे रहा है सर.
Dr. Charan.
Yes, sir. You are audible, sir.
Please, so very happy to have Dr. Bantaji and the first of the lectures. So now we are waiting the lecture of the Dr. Bantaji, and so continue with the third lecture, and excellent. You can say scientifically, most probably number of the students, they are the work on and wait, and as you know that Professor Y.K. Gupta ji very specifically mentioned that in those days most of the peoples, they are the moving toward the impact factor and moving toward the how we can increase the number of the publication. So now not only the, you can say, in India, this is the especially we need to the lot of work in a justified manner so that it can be helpful to the peoples, because we know that nowadays most of the agencies, they are the working on, you can say, the brain related or brain targeted system, because most of the people, they have the lot of problem like that. So nowadays this types of, this is the not only the related to the disease, but there is a lot of confusion and everything related to the brain. So for this purpose we need a justified work, and it will be the suitable for the society. So as I know that I am person of the pharmaceutics, and I don't know the more about the pharmacology, and not more than the, you can say, the technical things, which types of the going. But I know that in our institute, number of the person, they are specifically working on it. Dr. Charan is the working on the nanotechnology, and they have also testing the some of the product related to the brain target, so many things like that. So thank you very much, Dr. Tosharji, and I hope that if you have some of the space, then we can also do some of the collaborative work in the ISF College of Pharmacy, because our team, pharmaceutics as well as the pharmacology peoples, they are the quite sound, and they are doing the dedication. As we know that youngster in those days, they are the highly dedicated. So we have some of the space instrumentation also, but we have the nearby two very strong institute. One is the NIPER, another one is the UIPSP. Also help as and when we have the required. We can do some of the very collaborative work, and specifically it will contribute to the society. It will be the helpful to the each and everybody. So thank you very much, Dr. Charan, and continue with the third lecture, please.
Thank you, Dr. Thanks, Charan.
A wonderful talk on silicon nanoparticles in terms of the synthesis, its characterization, and the
आवाज break क्यों हो रही है?
So I'm able to
Charanji,
hear all of you.
your voice is the breaking.
So let me try and check it. So, uh, so you have mentioned a lot of studies in terms of the release, degradation, bioactivity of the protein lysozyme by loading it with the porcelain nanoparticles. And in a very lucid way, you have demonstrated the application of porcelain nanoparticles in imaging, as you said. This can be used for the delivery of anti-cancer agent, for example, temozolomide, which we have seen in the previous talk. So I hope we'll have the same sort of nanoparticles and the same type of carrier systems in real science and technology. And one more point, Dr. Tushar, I can remember the day when I was preparing these nanoparticles with you in the last in 29, early 2019 and 2018. So I can recall of those days. So thank you so much for having with us in the Ibro Spotted meeting workshop and on the posting. Thank you so much.
Thanks, Jen.
So for today's technical section, the next speaker is Dr. Vandita from UIPS Punjab University, Chandigarh. So, ma'am, I welcome you in this session. And for the audiences itself, so Dr. Vandita Kakkar is an assistant professor in the Department of Pharmaceutics, UIPS Punjab University, Chandigarh. She has a research experience of more than 10 years, and her area of research lies in the bioabsorption enhancement of hydrophobic drugs, especially we can say the BCS class 2 and 4 drugs. And she works in the area of development of the nano formulations of phytopharmaceuticals using some nanoparticle-based technology via oral as well as topical routes. And she also focuses on the scale-up of the nanoparticle production from lab scale to pilot stage, combating antimicrobial resistance, which is, I think, a huge challenge all over the world, and developing targeted delivery systems for cancer treatment. She has to credit 45 international or national research papers and review articles with H index 17 and more than 2100 citations. And she has 11 book chapters in international books, 78 magazine articles in Britain, South Asia magazine, and 4 national patent applications. Out of those, 1 is granted. And she has been awarded research grants around 0.62 million from UGC and Punjab University Seed Grant. And, she has 5 millions for BIZ Big BIREC grant, 2 millions from DST Sir, another grant of 2 millions from ICMR, and 7.2 millions Commonwealth Commission, UK. So she has industrial experience of 2 years, and she has to her credit several awards and accreditation, to name a few, as just a couple of seconds before. She has guided 16 postgraduate students, 2 project assistants, and 1 research associate, along with 11 UGC NRC training students, and presently guiding 3 PhD students at UIPS Punjab University. Also, she has transferred the research work technology to high-tech formulation, a pharmaceutical company based in Barni, Himachal Pradesh, and is consulting a research project of citrus byproduct. So, with these few words, ma'am, I welcome you all in the technical section for the day 2 in the post-coordinate meeting and workshop. And I like to have the presentation from your side. Thank you so much. Over to you.
Thank you so much, Dr. Chadan, for the kind introduction. And at the onset, I wish to thank Dr. J.D. Gupta and his team for organizing such a beautiful program of Ibro, wherein all of us, scientific community, we are sitting together, we are exchanging the ideas and the work currently which is being taken up for the brain research. And probably, as YK Gupta sir was pointing out, definitely there is a declining phase which we all can observe while we talk of the brain research, and we all need to collaborate, we all need to bug up with the areas on which we are working. so that we can reach the market. Being a pharmaceutical scientist, I feel that whatever we are doing, whatever work we are taking up, the main goal, the main mission should be that we are able to reach the clinic. We are able to reach the patient. Though we all are putting in all our efforts, we are into, most of us, we are working on nano part only. But when we talk of the FDA guideline, while we talk of the commercialization of any product, we have many, many challenges. And that is where we all need to collaborate. The point is where we can actually pitch ourselves and we can draft a guideline for the approval process. Now, when we are talking of any nano formulation, till now we have only a draft guideline which came up in 2007. Till now, we are in 2022. Still, we are working on a draft guideline only, and we do not have any final guideline while we talk of approval of any nano formulation. So, we are working so much. We heard Tushar, we heard Dr. Deepak Chitkara. They are working so much on nano. I myself am working on lipidic nanoparticle. Though I could transfer my technology of lipidic nanoparticle to the company for people, but that is for cosmeceutical purpose. That is for topical purpose, not for brain. Why we aren't able to do that? See, we are getting so much of funding for brain research. Now, the question here is that really we need to ponder upon, we need to think that what are the hiccups where we all can hold hands together and approach the authorities that we want this product to be there in the market. Help us to get the clinical approval, because we are doing safety, we are doing toxicity, we are doing everything, we are doing efficacy. Then why can't we come to the market? So I think this is a right forum where we all can take a pledge that we need to be together to get the approval for the product to reach the market. So with this opening remark, I'm going to, start with my work, my presentation, which I have taken up because Dr. Charan said, ma'am, your presentation has to be totally oriented towards the brain research. So yes, I did work on that. And I would wish that most of the students who are looking here, you must learn the preparation of lipidic nanoparticle, which is very, very important. What I have done is completed. But you people are going to really start with it today. You're going to hear to us. And I want all of you, if it is feasible, Dr. Charan, can we interact with them? No. Okay, but we can have queries from them, right? We can have questions from them. Let me know if it is visible.
Not yet. Yes,
Yeah.
it's presenting now.
Is it there?
Yes, ma'am.
Okay, perfect. So the title of my talk today is Delivering Curcumin Loaded Lipidic Nanoparticles to Brain, a POC study. Now, the purpose of this talk is to acquaint you that why I took up curcumin, why we took up lipidic nanoparticles, and how we could reach the brain, which was very challenging, because we have blood-brain barrier, which is a main barrier to cross any drug molecule into the brain for therapeutic efficacy. So the first purpose, because we have heard of curcumin a lot. Curcumin is the active molecule which is present, which is derived from turmeric, and the concentration of curcumin in turmeric. Turmeric is normal haldi, which we are using at home. And we all know that while we talk of this molecule, you talk of the therapeutic areas where it is working, whether it is cancer, whether it is any inflammation, whether it is heart disease, whether it is neurodegeneration. So this molecule exhibits pleiotropism, and it's working any area. You pick up this molecule and you try it, it is showing a therapeutic efficacy. So the first thing which came to our mind is that we have to pick up a nutraceutical molecule so that it can be given to all. Now, because we are talking of neurodegeneration, which involves Alzheimer's, which involves depression. So all these disorders, if we have something available in market, which can be taken up first as a prophylactic and then as a therapeutic, then it's going to be a revolutionary product. With this aim, we picked up curcumin, and because we know that this molecule has a very low bioavailability of 1%, so the main aim as a pharmaceutical scientist was that how we are going to blood that bioavailability, we want the drug molecule to be available in the blood for uptake. So the step which we took was that we are going to prepare lipid nanoparticles, and out of all the range of lipid nanoparticle, it was solid lipid nanoparticle which we picked up because we have a method to prepare them without using any organic solvent. We can target these very well at any site where we want. How? By countering the size of the particle, and that can be done by the method which we are going to produce. So I'm going to share with all of you the method which we used, which was very much scalable method and a very spontaneous, easy method, which you all can do it in your lab. You don't need much of investment for that. Now coming to the molecule, which is bis alpha beta unsaturated beta diketone, it's a hydrophobic molecule. As I told you that the solubility of this molecule is very, very low. It's 11 nanogram per ml, while the log P, it's around 2.98. So, the reasons of its low intrinsic activity were both of these parameters. That is, the solubility was very low. Then fast metabolism is there. And the when it is degraded, when it is just actually broken up, the metabolite also, they are not very active. Then the rapid elimination and clearance. And another problem with the molecule was it is not stable physiological pH also. So, considering all these challenges, we thought, okay, let us have something which is going to stabilize the molecule. Because if a molecule is not stable and we are preparing the formulation out of it, then also we are not going to lead the molecule to any final stage. So this DAY-2 molecule exhibits a very variable pharmacokinetics, a very low detectable plasma level because of the low bioavailability. And, the papers, they indicated that it is hardly 0.13 milligram in plasma after 15 minutes. And after 6 hours, if we observe the plasma, it is totally undetected. We cannot even see that. While if we see the IV data, we can see that curcumin at a dose of 10 milligram per kg, it gives a Cmax of just 0.36 milligram. While per oral dose, if we talk of that, was intravenous, and we talk of per oral, it is hardly 0.06 milligram, which is much below the therapeutic concentration which is required for producing any therapeutic effect. So the percentage of absorbed curcumin, it is going to remain constant, regardless of you are giving 100 milligram, you're giving 500 milligram, 1 gram or 8 gram. So if we talk of delivering curcumin to brain, we have different modes because I'm a drug delivery person. So it becomes very important that we must know that what are the ways we can deliver. So we have the local delivery, we have intranasal delivery, and we have the systemic delivery, wherein directly we inject at the site of the action. So while we talk of these delivery parameters, there were various ways we could have actually taken up a drug molecule or a therapeutic to reach the brain site. It could be via endogenous transporters, like we have the glucose transporters present on the brain, and then inhibition of efflux molecules, because most of the molecules which we want to deliver to the brain, they are higher molecular weight. So we need to inhibit those particular efflux systems so that we can aid the molecule to enter that site. Then we have the procedures for, the osmotic openings, or these are all the invasive ones, the blood-brain barrier disruption or the vasoactive mediated drug delivery, then the cell-penetrating peptide vectors, and the formulation approach. It is this part which I'm going to highlight and discuss with all of you, that how we formulated it, how we could observe that these nanoparticles, they have reached the site where we really want to. So when we talk of the barriers of the brain, we can see it is the blood-brain barrier, the blood cerebrospinal fluid barrier, and the blood tumor barrier. While we talk of the blood-brain barrier here, so it is the structural and the functional barrier, which is the main impediment, and it's this barrier which regulates the influx of most of the compounds from the blood to the brain, though formed by the BMEC, which is brain microvascular endothelial cells, the astrocyte, enphyte, and the pericytes. And it is very essential. We cannot sustain without a blood-brain barrier. For the normal function of the CNS, helps in regulating the molecules and responsible for all the metabolic activity. As I discussed with you, that it is equipped with specialized transporters like the glucose transporter for the transport of the essential molecule. So we can see here, we can clearly observe what all are there: the tight junctions, the microglial cells, the astrocytic foot processes, the pericytes, the axonal ending you all can see here, the endothelium. So how we just need to work on that, how we are really going to cross this and enter. So the gaps which are between the endothelial cells of the CNS, it is these tight junctions which we need to target, where we need to find the passage for the drug molecule to reach the brain. Then the pericytes and the endothelial cells, which are completely encapsulated by the basement membrane, the astrocytic and the feet, which envelop 99% of the vasculature. So if we just talk of the areas of the brain, the brain without the blood-brain barrier, we can clearly see it is the preoptic recess, the paraphysis, the pituitary gland, median eminence, the area postrema, and the pineal gland, and even the endothelium of the choroid plexus. So it is this, these are target sites where we really need to work on for passing across into the site, the target site where we wish to target the molecule. Coming to the various strategies which can be taken up for enhancing the bioavailability of curcumin. So we had the liposomes, the micelles, NLC, or it could be a combinatorial approach wherein we club curcumin with various adjuvants like piperidine or eugenol, because when we club these molecules with curcumin, they are going to enhance its bioavailability. They act as inhibitors, they act as flux inhibitors. Then we have SMIDs and phospholipid complexes, the polymeric nanoparticle and the solid lipidic nanoparticle. So we are going to take up the solid lipidic nanoparticle, and the main, the advantages of these lipidic nanoparticle actually prompted us that we must try them for preparation of a targeted delivery system for the brain. Because there is no solvent which we are going to use here, it's going to help in controlled drug delivery, has high drug entrapment, improved drug targeting, and then the most important thing, that stability is very good. There's no aggregation as we observe in case of liposomes and even some of the polymeric nanoparticle. So the preparation of these lipidic nanoparticle, which we have taken up, though we have several methods: high pressure homogenization, cold pressure homogenization, many other sophisticated techniques are available. But today I'm going to talk about a very simple technique wherein we do not need any investment at the start of the project, where we can start preparing these nanoparticles on the first day. So it is the microemulsification technique, and we all know that these microemulsion, they are very clear, they are thermodynamically stable and optically isotropic system which require much less energy, and they are obtained very spontaneously by mixing the surfactant, the co-surfactant, lipid and water. It is very important here that I must highlight that the microemulsion, as we are mentioning here, microemulsions are clear. Now, the word microemulsion, we all know, it's a misnomer. The particle size of any microemulsion is in nano. It is below 100 nanometer. So while it's going to be below 100 nanometer, it is going to very, clearly show that it's going to be stable and clear, transparent. If the microemulsion is not clear, if it is not stable, then believe me, we cannot have lipidic nanoparticle which are going to have a nano size. So the first step wherein you are able to obtain a microemulsion which is very clear, which is very transparent, indicates that you are moving with the concentration of surfactant, co-surfactant and lipid in a very right and a perfect ratio. In case you are not using the optimization technique. If you're using the optimization technique and you're preparing your ternary diagram in accordance with that, then definitely we are going to get a microemulsion which is very, very stable. So in the aqueous media, these lipidic nanoparticles, they are going to be formed while you are going to pour them in cold water, which is maintained at 2 to 8 degrees Celsius. So as I just discussed with you, that we need to have a very optimum system. The concentration of surfactant, lipid, co-surfactant and water is very important. Now here I wish to emphasize one very important thing, because it's been almost 12 years I've been working with the lipidic nanoparticle. One mistake which we generally make while we are optimizing the formula is we keep on adding the ingredients to see that the system is going to be stable. Once you get a very clear system, after preparation of your lipidic nanoparticles, you come to know that the concentration of surfactant and co-surfactant which you're using, they cannot be given orally. It is very important at the first go, you need to check the FDA IIG limits so that you know that the percentage of any surfactant, whether it is 2-in-80, whether it is phosphatidylcholine, whether it is PVP, whatever the lipid is, whatever the co-surfactant is, we must know that they are within the oral limits. If you are preparing it for oral, you must know that they are under the limits mentioned by FDA IIG. Because once you have prepared the formulation, reverse engineering in case of lipidic nanoparticles, once you have formulated your final formulation, is not going to be a very easy task. So it is my submission to all of you that while you start your protocols, you must keep in mind considering the FDA IIG limit for the particular intended route for which you are preparing your lipidic nanoparticle. Coming to the method of preparation, a very easy and a spontaneous method. We have the aqueous phase and the lipidic phase. Both of them, they are melted at the same temperature. And then we are going to pour the aqueous phase into the lipidic phase to prepare a clear microemulsion. So once we get a clear microemulsion, we are going to pour that clear microemulsion into the water which is kept at 2 to 8 degree under continuous stirring for around at 5000 rpm for around an hour. So this microemulsion based system is a two-phase system which is composed of an inner and outer phase, and they are produced by stirring an optically transparent mixture at around 65 to 700 rpm, and they are characteristically composed of low melting fatty co-emulsifiers and water. And this hot microemulsion is dispersed finally in cold water, and droplet size, as I told you, it is already contained in the microemulsion. So this name microemulsion is a misnomer, and therefore no energy would be required to achieve that nano size which we are talking of here. Now here is a small video to help you see how we are preparing the nanoparticles. Is the video visible? Yeah. So you can just have a look that how these lipidic nanoparticles are being prepared. Now we have modified the method a bit, in which we have started with the high speed stirring. So rather than taking the magnetic stirrer and a mechanical stirrer, we are using the ICA high speed homogenizer. By using that, we have been able to reduce the time of stirring by almost 50 to 60%. So if we are preparing a batch of, let us say, 500 ml, so the time which previously was a bit, around 1.5 to 2 hour has reduced down to 40 to 50 minutes. So if we can have that ICA high-speed homogenizer, we can place the final lipidic nanoparticle solution, that is, which we are the final stage wherein we are pouring the hot microemulsion in the cold water, and we can give 2 cycles of 20 minutes each to have a reproducible particle size in that. So it's very good if we can really go in for ICA homogenizer after using the magnetic stirrer. So after the weighings has been completed, both the aqueous phase and the lipidic phase, they are kept at the lipid melt temperature, 10 degree above the lipid melt temperature. So we have here all the 3 phases, the lipid phase, the co-surfactant phase, the water phase. So you can see both the phases, they are being melted on a 6-stage homogenizer. And here we have the clear microemulsion which is being formed after the addition of the aqueous phase into the lipidic phase. And this is the ICA homogenizer. So it is kept in an ice bath so that the temperature doesn't rise, and we can have the lipidic nanoparticles which are formed with a very, a regular shape, and all the lipidic nanoparticles, they are reproducible with respect to the size and the morphology. So that was a brief. So these are the lipidic nanoparticles, and then we refrigerate them until we are going to move ahead with the characterization of these lipidic nanoparticles. So there were many challenges as I talked to you, that stability, the drug entrapment. So while you're working with these parameters, you have to be very specific about the addition of your drug to either aqueous or the lipidic phase. Most of the drugs, they are not soluble in lipid phase. And we unavoidably, we add the drug to the lipidic phase. Now, while we are adding the drug to the lipidic phase, we could observe that the drug entrapment and the total drug content which we were getting, it was much, much below as compared to the one in which we are adding the drug to the aqueous phase. So this is very important parameter that we must channelize, we must analyze first that what is the basic characteristic of drug, whether you're going to add it to a lipidic phase or an act, because most of the times we tend to add it to the lipidic phase, and with that we do not get the entrapment, because while you are adding it, there are drug losses as you convert, as you pour them into your final solution, that is the water which you have kept at 2 to 8 degree. So, and another characterization of excellence is a serious challenge because of the colloidal size of the particle. So here you have to optimize the speed which you are going to use for the ICA homogenization technique. Another challenge was that how much time you are going to give at every stage. So while we increase the time from 10 to 20 minutes, we could observe that the particle size is drastically increasing. So it becomes important that you optimize the method with respect to the speed. So what we could see that it is, during the first 10 to 15 minutes, not even 15, 10 to 12 minutes, while you're going to pour your hot microemulsion into the cold water, you must give around 12 minutes at a speed of 12,000 rpm. Thereafter, give a break of 2 minutes, and then for next 10 to 15 minutes, it has to be around 8,000 rpm. So we could see that there is reproducibility in the particle size which we are getting. The morphology of the particle is uniform. So these all challenges, you must keep in mind before you start your work. So these were the results which we could obtain. A very good particle size of around 75.3 nanometer, and the TEM study also, it confirmed that the particle size is of the same range as we could observe in case of the Beckman culture PCS technique. DAC and IR confirmed the lipidic nanoparticles. They were formed because there was disappearance of the peaks, and we could see new broader peaks here. And XRD revealed that the lipidic nanoparticle, they were amorphous in nature. We could attain a very good drug content of 93.7% and drug entrapment of 84%. In vitro release was prolonged up till 7 days, which is a very good time if we are talking about even an oral drug delivery system. The PK study, they were done in rats using the mass spectrophotometric studies, and we could see that we have taken up various doses of these lipidic nanoparticles, starting from 1 milligram, 12.5, 25, and a very high, that is the 50 milligram per kg dose, which is compared to the free curcumin. And we could see that even the small dose of 1 milligram per kg could result in a very good Cmax as compared to that of the free curcumin. So the results, they indicated that the AUC for CSLN was 39 times higher at 50 milligram per kg dose as compared to that of free curcumin, while the increased bioavailability we could observe at 1 milligram per kg was 55 times. It was 59 times with 12.5 and 32 times with 25 milligram per kg dose when it is being compared to the free curcumin. Again, the volume of distribution also, it was 53 times lower, because we are talking here that the retention was much better as compared to the free drug. So these all the result, they indicated a prolonged circulation time of the lipidic nanoparticle with significantly lower clearance and the volume of distribution. Then we tried testing these, and because we are talking of all categories of cancers, so we tried testing them in various cancer cell lines, and we could see that, in the MTT assay, we could observe significantly higher apoptotic activity was there with curcumin nanoparticle on HL60 cell line as compared to that of the free curcumin. So we all can see the concentration here. The IC50 value of CSLNs is only 1 microgram vis-a-vis the curcumin concentration, which is 7. So all these studies, then we did the DNA fragmentation, and we could observe that the lipidic nanoparticles, they could produce a much better DNA ladder, which is very important while we talk of the penetrability of the lipidic nanoparticle inside the cells. Then talking about the G0 DNA fragmentation, which comprises of the apoptotic and the debris fraction. So we could see that the lipidic nanoparticle, they could produce a very good percentage of this. So, in the control cell, it was only 2%, while it was 96% with lipidic nanoparticle when they were treated at a concentration of 10 microgram per ml. And this very emphatically, they reveal the potential of lipidic nanoparticle to bypass the barriers and enter the cell to prove its therapeutic efficacy. Then talking of the loss of mitochondrial membrane potential, again, the HL60 cell exposed to nanoparticle and free curcumin for 1 hour at different concentrations of 1. So you can see, the concentration of lipidic nanoparticle already which we are using is 10 times less here. And we could see that CSLN, they could cause the mitochondrial damage and decrease the mitochondrial membrane potential by 44% in comparison to free curcumin, which showed only 9% decrease at the same concentration. So this was a very important finding. Then these lipidic nanoparticles, they stimulated the caspase activity, which is very, very important for the apoptosis. And these CSLN, they produce a dose-dependent increase in the activities of caspase 8-9 in HL60 cell. And the effect was definitely 3 times more as compared to that of the free curcumin. Then on the expression of important proteins, which are again involved in the initiation of apoptosis. So they showed significantly better suppression of the NF-kappa B and significantly higher marked fraction of the cytochrome C, which was released in mitochondria with nanoparticles as compared of the curcumin. We could see here cytokine CTNR1 is one of the most important potential activator of NF-kappa B, where the constitutive activation of NF-kappa B is mainly assigned due to the autocrine expression of this TNF-R1. So we could see the upregulation of this also with the nanoparticles. You can see the doses here and you can see the expression out here. So, these studies, they all indicated that it is having a very good potential to be clinically translated, and it can be given a status of a therapeutic because as of now, while we talk of curcumin, it's a nutraceutical. We cannot treat it as a drug. It's not being treated as a drug. But looking into the potential therapeutic efficacies, it is reflecting in any studies, whether you're talking of the cancer studies or you're talking of the animal pharmacodynamic in vivo studies, we can definitely say, that this molecule can be taken up as a therapeutic in the market. Then in the pharmacodynamic evaluation, we performed an Alzheimer's model wherein we could observe that, given this lipidic nanoparticle, we could see that the latency time decreased significantly as compared to the positive control and was very similar to that of the naive control with all the doses, though SLN50 could produce markedly lower levels, even below that of the naive control, revealing very clearly that the nanoparticles could reach the brain upon oral administration, which was a very, very important finding. And the biochemical estimation also very clearly indicated that the curcumin lipidic nanoparticles, they could reduce down. While we talk of the MDA, it could decrease all these levels while we compare to the positive control. So we can see, we can observe that, see, these FC is the free curcumin group, in which lipidic nanoparticles were not there, but we have solubilized the curcumin at a dose, a very high dose of 50 milligram, and then given to rat. This is a group of blank solid lipid nanoparticle, wherein we have prepared the lipidic nanoparticles, but we have not added the drug to it. Similarly, you can see the effect on the SOD, the catalase, acetylcholinesterase, and the GSH, which was increased comparatively, and it was almost, we could, though it was not equivalent to naive, but there was no significant difference amongst the group of SLNs with that of the naive control. And the histopathological studies clearly indicated that, the rats, they could heal in case of the Alzheimer's model of which I'm talking about here. And we could clearly identify that these nanoparticles, they have reached the brain and they have shown their therapeutic efficacy. Again, similar studies, there was another model of depression on which we have evaluated, and we could see that the immobility time, it reduced significantly while we were giving the solid lipidic nanoparticle as compared to that of the free curcumin dose, which was very similar to the dose of the lipidic nanoparticle, but the results with free curcumin was same as that of the control. We couldn't see any much significant difference by administration of oral free curcumin. Coming to the proof of concept study, so it becomes very important while we are observing the therapeutic effects. How do we see that, in reality, your lipidic nanoparticle, they have reached the site where we are talking of? So we conducted these studies, the confocal microscopic studies, the biodistribution, and the gamma scintigraphy. And in fluorescent and confocal microscopy, what we have done is that we have taken the group, the control, and the lipidic nanoparticle group, wherein we have given the free withdrawn animals, they were sacrificed, the brain was harvested, and we could observe the brain sections under the fluorescent microscope. So you can see the lipidic. Now this curcumin, it exhibits fluorescence. That was an added advantage, and we could very clearly see, you can see these yellow spots. So these clearly indicated that, the nanoparticle, they have reached the blood, right? So this was in plasma, SLNs in plasma. And this is the picture of the blank plasma where we couldn't see any fluorescent molecules as we could see in SLNs. Similarly, in the brain cryosections, we could see that, SLNs, we can see the site and we can see the lipidic nanoparticle present there, and that too in a very dense concentration, which has clearly indicated that the nanoparticle, they could bypass the RES and could bypass the blood-brain barrier and reach the brain. So these particles, they confirmed their effectiveness. They could cross the gastric mucosa, and it could be concluded that these SLN, they maintain their integrity in the plasma and pass through across the blood-brain barrier in intact form. Then we performed the biodistribution studies, and first step was that we had to see that they are stable with the labeling which we performed with technetium-99. And then we performed these biodistribution study in mice where we gave all the animals, again, the free curcumin, solubilized curcumin, and then the lipidic nanoparticle. And we could observe in all the organs the percentage radioactivity which was present as compared to that of the free curcumin. So we can see in brain, which is blue color, that is the one, in first R, the C-SLNs, they could produce such a big, huge activity while we compare that to that of the free curcumin. So that was our main, target, that whether we are able to reach the brain in sufficient concentrations or not. So the method was after this, we performed the gamma scintigraphy in New Zealand rabbits, and all these studies, they were performed in DRDO, New Delhi. So the animal New Zealand rabbits were taken, and after per oral and IV administration of the radio-labeled curcumin, lipidic nanoparticle, and free curcumin, these animals, they were anesthetized by ketamine intramuscularly, and then they were fixed on the board in posterior anterior position, and then imaging was being done. And here are the images, the gamma scintigraph of the animals, of the rabbits, and we could clearly see that after oral administration also, we could see lipidic nanoparticle in the brain area. Well, this is for IV, where again we can see a huge concentration here. And with oral also, comparative concentration, we could observe in the gamma scintigraph of the rabbits, the New Zealand rabbit, which we have tested. So the summary is that in this study, the AUC value for C-SLN, it was 8.135 times greater as compared to that of the free drug. And the ratio of the AUC for C-SLN IV versus that of free drug in blood, it is less than 1, while the ratio in brain is very promisingly, this indicates 30 times higher and preferential distribution of the drug from lipidic nanoparticle into the brain tissue. And similar concentration, similar observation, we have seen the gamma scintigraph, they were observed for the oral as well. And this was for the first time we performed this study and reported it, and we could publish it in a very good journal. Again, the purpose is not only publishing, but it is to have the product in the market. So the highlights are the improved plasma level with the curcumin lipidic nanoparticle, which was attributed to the particle size, which was very low, which could bypass the RES, and definitely free curcumin because it's undergoing the metabolic transformation. So the bioavailability is very, very less. And the surfactant and the co-surfactant, they have contributed to the high permeability. So we could see that all these nanoparticles, they were intact, they were not degraded. So the system, the solid matrix which we have used is apt for preparing the lipidic nanoparticle, and the prolonged circulation and reduced clearance we could see very clearly. Passive uptake, we have assigned it to pinocytosis and endocytosis, and also we mainly attributed to the active uptake by the brain endothelial cells. And this all is attributed to the adsorption of the circulating plasma apolipoprotein E on the surface of SLN, because we have on it the coating of the surfactant, which is polysorbate 80, which is selectively taken up by the ApoE receptors, which are present on the blood-brain barrier. So that was all about my presentation. Here are a few of the publications, the recent ones. And after curcumin, I worked on tetrahydrocurcumin, and we have got some grant for preparing the topical formulation of this molecule. So we had a technology transfer also for the topical formulation, for the commercialization of the product. And the patent was granted last year to me. So these are a few awards and achievements. So thank you so much for the patient hearing. So I think sometimes later becomes never. So we all really have to do it now. So thank you.
Thank you so much, ma'am, for a wonderful talk on the basics of the drug, that is curcumin, this is class 4 drug, ma'am, having solubility as well as permeability issues. It's very difficult to form a carrier system of the same drug. Most of the drugs are this is class 2, having only one issue associated with solubility. So you have taken the presentation from very stressed to the in vivo evaluation. Clearly shown the data about the synthesis of application of the excellence. And one of the most important point I would like to highlight is the concentration of lipids. Because we more of first was the taking of more lipid into the SLNs for having a high dependency of the drug load. So which is an important point in the presentation. Followed a lot of studies in terms of cell line studies. You have performed the gas-based active mitochondrial potential genes. Brain studies. And lastly, we have shown the proof of concept studies, wherein you have shown the confocal studies. We can clearly see yellow color also the presence of the same drug molecule in the formulation that the particular site of action. So overall, it is an informative like the first one pursuing the regulatory science and in terms of and pharmaceuticals. So thank you so much. And it is interesting to hear you after a couple of years back. And thank you so much for the post-birthday meeting workshop. ISFCP. Thank you so much.
Thank you so much, Charan, and thank you to Dr. Gupta for giving this forum to interact with you all.
Can I ask a question? If there's
Definitely. Yeah.
If there's time, yeah. The question I had is, it's very interesting work and very extensive. Thank you. And then your presentation is very detailed on that as well. Most people don't do that, myself included. So thank you. The question I had is, you did oral gavage or oral delivery. At one point, you mentioned that you solubilize pure curcumin, right? Not in SLN, but something else. How was that done? Was it in a solvent, so co-mixture of solvent with PBS or something, or was that done?
Yeah. Okay. So what we have done with free curcumin is that we have taken in all these system, we didn't prepare lipidic nanoparticle, but we have taken the surfactant and the co-surfactant in which your curcumin is going. So that's why we have mentioned it as solubilized curcumin.
Okay.
So that we could observe that it is not because of the solubility here, but it is because of the lipidic nanoparticle that it's going to enter the brain.
Yeah. One other thing would be, like, since you've already done, in vivo studies, we've tried similar things with a bunch of hydrophobic molecules. In vitro, we see really great data, but for some molecule, what tends to happen is we don't see them permeating in vivo. But when we functionalize our particles with different surface moiety, we get them to go from mucoadhesive to mucopenetrating. Is that something that you've tested for your, lipid-based systems as well? Like, have you done any confocal images of the GI tract cross sections?
Tushar, I think you're very right. While we talk of the ligand-driven system, I think they are very good. But in case of our lipidic nanosystem, it was a finding that because we have used tween 80, that tween 80 helped us, it acted as a ligand here. And it helped us to permeate because of the apolipoprotein E receptors which are present across the blood-brain barrier. We could see that preferential uptake because of that. So definitely we can have, we can decrease down the concentration of tween 80 or surfactants here, and we can have vegan in place. Definitely, we can do that.
Perfect. Thank you.
Thank you. Thank you so much. I think Charan has lost the connection.
Can you hear me? Okay.
Yeah, Charan.
Thank you, ma'am. So,
Okay, Charan, thank you. Yeah.
for the next talk, we are having Professor Pankaj Seth with us from National Brain Center, Manesar, Gurgaon, Haryana. So, sir, please accept my sincere regards to you. Yes. So, I can recall the moment when I had an opportunity to interact with you at Ibro School in 2017, honestly. So, from today, I was trying to get connected with this. So, with the help of you in between these 5 years from the for the various applications in Ibro, I seek your consent as well as help, and with your motivation, sir, I was involved in a lot of applications. So, uh, for today's talk, like just give me a couple of seconds, sir.
Just confirm, Charan, if you could see my slides.
Yes, sir, I can see the slides. Sir.
Okay, thank you. So, shall I start?
Sir,
Shall I start? I said, with your permission.
sir, can you please click on the hide option in your presentation?
Sorry?
Yes, that's fine. So, sir, can you please allow me for 1 to 2 minutes for the introduction?
Okay.
So, thank you so much, sir. So, participants, today we are having Professor Pankaj Seth from NPRC, Manesar, Haryana, with us for a technical talk. So, Dr. Pankaj Seth's research endeavor after returning to India from NIH, National Institute of Health, USA, has been originality of thinking, taking up challenges of direct relevance to the national health problems, particularly HIV-1, drug abuse, Zika virus-mediated neuropathogenesis, and very recently, effect of SARS coronavirus 2 on human brain. Recently, he started research to understand why COVID-19 patients exhibit neurological symptoms as brain fog. In fact, Dr. Seth had exposed this possibility in early stages of the global pandemic April 2020 itself. In a comprehensive review published by him, stating that SARS coronavirus 2 is more than a respiratory virus, which is published in ACS Chemical Neurosciences 2020. That is now accepted worldwide in a consonance with his vision and is being widely cited globally. With these capabilities and expertise, Dr. Seth's laboratory is geared up to understand the cellular and molecular mechanisms as to how the SARS coronavirus 2 affects the functioning of brain of COVID-19 patients. His research has been highlighted twice by Nature India and by the International Society of Neurobiology, based in USA, electronic and print media reflecting its impact in the field. Dr. Seth, because of his passion to understand national problems which are neglected by others, he earned a distinct reputation among his peers globally. Despite the fact that India has millions of HIV/AIDS patients, less than half a dozen researchers are engaged in studying molecular mechanism of neurocognitive and motor deficit, and those are known as neuroids in such patients. As it is challenging, Dr. Seth overcome these challenges with diligence, vision, and a scientific approach. Additionally, he was invited as collaborator by researchers from Johns Hopkins University, NIH, University of Nebraska, and other prestigious institutions globally and within India. His research NPRC has been extensively funded by NIH RO1, R21, Office of AIDS NIH, and these are super competitive grants. I hope the audience knows about it. And these are funded by US India Bilateral Collaborative Research Partnership, that is CRP, on the prevention of HIV, AIDS, and comorbidities. Apart from this NIH grants, his lab is well funded by DST, DBT, and ICMR. Today, his research laboratory is among the very few laboratories in the world that has developed and validated a well-characterized model of primary culture of human neural stem cells to understand brain disorders. The National Academy of Sciences, India, the National Academy of Medical Sciences, and Indian Academy of Neurosciences, IN, have elected him their fellow. He has been appointed as an area expert in number of DBT, ICMR, task force to lend his expertise. Moreover, he also serves on editorial board of numerous international journal in various capacities. Lastly, Dr. Seth serves on board and councils of several international scientific bodies, reflecting his research accomplishments and international visibility in the field. He is currently serving as board member of International Society of Neurobiology, that is ISFCP, USA, International Council member, Society of Neuroimmune Pharmacology, SNIP, USA, and as council member of Asia Pacific Society of Neurochemistry. He is promoting the cause of neuroscience in developing countries. Within India, he made significant contribution for promotion of neuroscience with his active role as General Secretary of Indian Academy of Neurosciences. So we are happy to have you, sir, in the technical session for day 2 of Ibro's board meeting and workshop. And this is a proud moment to me to introduce you, sir, in this conference and the national as well as international platform virtually. Thank you so much, sir. You please, I request you to please go with me.
Thank you, Charan. Thank you so much for that kind introduction. I had sent you that one-page write-up, hoping that you will read some of it. One-page write-up was just to ensure that you include me in this important meeting of yours. And I'm glad and I'm very thankful to all of you, the organizers, to give me a slot to talk about some of our research which we do at National Brain Research Center. So National Brain Research Center, as you would know, is located in outskirts of New Delhi. Can somebody mute? There is some lot of background. Yeah, thank you. So it is situated out at the outskirts of New Delhi, around 40 kilometers from New Delhi airport, and is destined and designed to carry out understanding how the healthy and the diseased brain works. So at National Brain Research Center, as we all know, inquisitiveness is very important for research. The researchers at National Brain Research Center are working on various aspects of brain disorders, starting from learning and memory, auditory cortex, how we see, how we learn, and of course, one group is also working, or two groups are also working on neuroinfections, and we are one of them. The National Brain Research Center has also been designated as an Institute of Excellence by the Government of India for its capacity as well as expertise in brain research. Now, considering that the audience is consisting of a lot of students as well, I thought I will make my presentation a bit simpler and highlight some of the research what we have done and also emphasize about how research is to be done. So as was emphasized by my previous speakers, Tushar and then Dr. Kakkar, and as well as Dr. Gupta, who had emphasized that it is the collaboration which matters a lot. And gone are those days when people used to work in their silos. And today, the research is done in a lot of collaborations across the disciplines, and that is required because research is like a multi-piece puzzle, And each one of us come with our expertise and put one of the pieces together to make a complete picture to help us understand the normal and healthy functioning of brain, let's say, which requires both passion, patience, and of course, sustained funding support. Now, to find a treatment for a disease, usually the research is bidirectional. Number one, we as scientists or basic scientists work in our benches and try to understand the normal functioning of, let's say, brain and brain disorders, and then try to see and relate it with the patients. The other way is also possible, like we just saw in this recent pandemic, when people were having problems of respiratory disorder and even brain fog and brain disorders, the clinicians reached out to the basic scientists to figure out what this novel coronavirus is about and how does it affect the brain cells and the mucosa membranes and causes the cytokine storm and so on. So research is considered both from bench to bedside and bedside to bench. And it is only when we work in tandem together, we can actually be, as was shown in the recent example of the success of understanding the novel coronavirus, designing drugs and vaccine against it, because both the clinicians and the scientists work together. Now, for the topic of my talk today, that SARS affects the brain cells also, and it is causing a neurological disorder in long COVID patients. Now, if as a researcher, what your students should be thinking about, and all researchers should be thinking about, how viruses affect human brain. And so the first question which should come to our mind is, can viruses actually reach brain? And if they can, what are the different mechanisms by which they can? So, and once they reach into the brain, what cell type do they affect? What decides the tropism or liking of particular cell type by the viruses? And if it attacks a particular cell type, how does it affect the cellular machinery? And if we can understand these steps, we can actually help in delaying or controlling or curing these disorders. So since we're talking about viruses and we're talking about brain, it comes to the point that we need to understand something called as neurotropic viruses. Now, what are neurotropic viruses? As the name itself is telling you, the viruses which have attractiveness towards nervous cell tissue are neurotropic viruses. And this term is usually described for the ability of the viruses to infect the nerve tissue. And if you look at these list of the viruses which I've listed over here, and as you go from top to bottom, you will see I've increased the size with the fact that perhaps you would have heard these names much more as compared to the Venezuela equine encephalitis, which is also a neurotropic virus, Japanese encephalitis virus, which affects a large portion of our northern India, where you have a lot of kids which are succumbing to the Japanese encephalitis virus, Nipah virus, West Nile virus, Zika virus, of course the rabies virus, and to some extent HIV virus also has been known to be affecting the brain tissue. And I'll maybe talk to you about that some other day. But to this list of neurotropic viruses, which is obviously not complete, but recently the SARS-CoV-2 virus is also said to be affecting the brain cells. But is it really a neurotropic virus? Is definitely a big debate. But definitely what is been confirmed now is that SARS-CoV-2 virus does cause damage to the brain cells. And that is what is the topic for the day. Now, for viruses to be neurotropic and then invade the tissue, brings to a property of these viruses called as neuroinvasion. Again, the word itself is telling you the invasion of the nervous tissue by the virus is basically neuroinvasion. And this depends to quite an extent on the receptors that pathogen uses to infect cells. For example, most of the viruses are receptor dependent. Let's talk about HIV. HIV requires CD4 receptor and a chemokine co-receptor, either one of them, such as CXCR4 or CCR5. So along with CD4, it requires one of these chemokine co-receptors to get into the nerve cells or brain cells, I should say. Zika virus also causes microcephaly and it uses AXL tyrosine kinase receptor. Now, SARS-CoV-2, I guess, is a virus thanks to the popular media, the animations which you have seen, the pandemic which was, has impacted the population around the world. It has also helped us to understand at least SARS-CoV-2 how it does. All those animations which we saw during on the television during lockdowns and other places clearly indicates how SARS-CoV-2 virus, the novel coronavirus, binds to the ACE2 receptor and uses the TMPRSS2 as a co-receptor, and that helps the virus to get into the cells which it infects, particularly all the ACE2 positive cells. As far as the brain is concerned, the nerve cells or the neurons usually do not have the ACE2 receptors, or as compared to their presence in the systemic system as well as the lung tissue. Neuropilin is said to be neuropilin 1 is said to be the receptor by which the SARS-CoV-2 is actually affecting the nerve cells. Now, currently in our laboratory, we are working on 3 viruses. We started working with HIV once I returned from NIH. And the reason why we're working with HIV was because it causes something called as HIV-associated neurocognitive disorders, which basically simply means that the HIV/AIDS patients have certain kinds of neurocognitive deficits, which is dementia-like symptoms, which is not as severe as dementia, but it does have, brings in neurocognitive deficits in these HIV/AIDS patients, which is noticeable. And that is one of the morbidity in HIV/AIDS patients. Now, Zika virus, on the other hand, is another virus which has neurotropic abilities, and it specifically works on the neural stem cells because this is a developmental disorder virus. What this virus does is actually it passes through the placental barrier while the infant or the fetus is in the utero, and the in utero infection of the brain cells, particularly neural stem cells, occur during the first trimester of the infection of the mother during her pregnancy. And it is so clear that it works only through in the first trimester or the first 3 months of the 9-month pregnancy of the mothers. And if the mothers get infected those first 3 months, there's 7 to 8% chances that the kids which are going to be born to these mothers which were infected in their first 3 months of pregnancy may develop microcephaly, which is basically abnormally small head size of these kids. These kids have very poor outcome in their lives because their brain had not developed, and that happens because when the Zika virus infects the neural stem cells, it interferes with the development process because the majority of the cell division and formation of different brain parts is happening through the proliferation as well as differentiation of these neural stem cells. And all that is affected by the presence of the virus. And this we can again talk in great detail in some other day. So today the focus of the lab, of the talk is on the SARS-CoV-2. And the reason why I chose this one is because not much is known about SARS-CoV-2 and its effect on brain, which is been studied because a lot of patients, especially the long COVID patients, the COVID-19 patients which had longer stay in the hospitals or took longer to recover from the SARS-CoV-2 virus, are the people who are developing brain fog. I'll define what brain fog is in subsequent slides later on. For all these 3 viruses, we have been using a robust model system of these human fetal brain-derived neural stem cells. So why did we start with fetal brain-derived stem cells, which is a very, very difficult model to do, especially if this is a primary in origin, which means that these are immortalized cells. So what we do is basically we derive human neural stem cells from the early gestation, from the aborted tissue, which we get from the hospitals. And once we get them from the hospitals after a informed consent of the mothers and following all the ethical guidelines laid down by DVT and ICMR, we then bring these tissues into our lab, we dissect them, we isolate the brain parts and grow them in culture in well-defined media. And all this is very well published by us. And if anybody is interested to learn this technique or to know more about it, we'd be happy to give another talk and invite them to our lab to come and see how these neural stem cells are cultured. Now, the beauty of these neural stem cells are that they grow both as monolayer, as you can see the brain part giving out, sprouting out some neural stem cells in culture, which is poly L-lysine coated plastic over here. They grow as beautiful monolayers, highly proliferative cells, or as 3D cultures. You can also grow them as suspension cultures. And what you see over here in A, B, and C is the increase in the size of these neurospheres, which are like perfect sphere-like structures and other properties of the neural stem cells. When we characterize these cells for their stemness, we observe that they have all the markers. Three of them are shown over here for neural stemness. One is nestin, which is a neural stem cell marker. Another is SOX2, which is also used in the iPSC technology by Yamanaka, one of the Yamanaka factors, a transcription factor, which is required by the neural stem cells. And of course, the proliferation marker Ki67, also used in cancer biology, to see that these stem cells are indeed stem cells. And we have, using this model system, we have published several papers from our lab, as well as in collaboration, two of which shown in blue over here were featured in the Nature India, by Nature India as well. Now, the beauty of this model system, as I said, since they are neural stem cells, they can proliferate as well as differentiate. Now, if you look at how these spheres are formed, let's try to look at this time-lapse microscopy, and I will show you as to how one neural stem cell is actually divided into 2, and then into 4, 8, 16, and so on. And within 48 to 72 hours, you will see that one neural stem cell actually has given rise to these multiple neural stem cells, which forms perfect sphere-like structure. And we use this model to, again, this property of these neural stem cells to make neurospheres by adding certain viral proteins to see whether the diameter which is formed with and without that protein or that test agent is affected in the same time points. We can also disrupt this, bring them to single cell, and count the number of cells which came out from one single cell, and then also figure out how the proliferation is occurring and whether it is being affected by the viral protein or not. Similarly, so the second property of these neural stem cells is differentiation. What you just saw was proliferation, and now you're looking at differentiation. Now, if you look at this picture over here again, here is a neurosphere which was differentiated into from neural stem cells into neuron. So this went under the process of neurogenesis. And what we're looking at different frames of pictures which we have taken out of these neurospheres. If I can draw your attention to this green cell over here, is showing you a cell. Basically, it is marked for TUJ1, which is an early neuronal marker. You can see that the neural stem cells which are undergoing neurogenesis is giving one beautiful cell over here. This higher magnification picture also then further tells you that you see a lot of green cells which are indicative of formation of new neurons. Blue obviously is the number of cells, which is indicated by the number of nuclei shown in blue over here by stained for DAPI. And the cells which are shown in yellow and orange are basically neural stem cells marked for the nestin with Texas red. And the yellow and orange color indicates that they are still under the process of differentiation, whereas the green cells indicate the differentiation process being completed and formation of new human neurons out of those neural stem cells. So these two properties of neural stem cells have helped us to understand a lot of disorders or understand how HIV, Zika, and then now SARS is affecting the brain cells. So the potential of these human neural stem cells is immense. And we have, using this stem cells model system, we have studied the effect of HIV on stemness of human neural stem cells. How we study this? We take the neural viral proteins of HIV, such as STAT, GP120. We can add it onto the neural stem cell culture and then look at whether the cells proliferate in the same rate or that rate goes down or is increased. So obviously these viral proteins are bad for the neural stem cells. So what we found was it basically reduces the rate by which they proliferate. We also looked at the effect of HIV on neurogenesis simply by adding the viral protein when the cells were differentiating into neuron to see whether at the end point do we have enough neurons being made from the same neural stem cells as per versus control. Now, since astrocytes have a very important role in having the functions of neurons, we have also looked at effect of HIV on astrocytes. We have looked at effect of HIV on glia-mediated neuronal damage and Zika viral proteins as well on neural stem cells. And very recently, of course, after the pandemic, starting from 2020, we have also started to use this human neural stem cell model to understand what are the mechanisms and what are the viral proteins of SARS-CoV-2 which are causing neuropathogenesis in an attempt to understand how brain fog is occurring in these patients. Now, a little bit about coronaviruses. We all know bats are the carriers, and usually after the bat, there's an intermediary host also. What we have learned from SARS and then MERS and then novel coronavirus, that the years by which it is taking to bring the new pandemic is reducing. We don't know when it will be the next pandemic. Hopefully, it will not be as bad as COVID-19. But what is interesting to see that you always had an intermediary host, and then you once it comes into the human system, it is very, very important to understand the degree of it. Now, why our lab or the research group in my labs thought that this COVID-19 can be more than a respiratory viruses, because we had learned from earlier literature that SARS and in MERS, the other sister viruses of this novel coronavirus, there was some involvement of brain tissue. And that is why we always believe, while the whole world was worried about COVID-19 and its effect on the lung and as a respiratory virus, we right from the start, after the break out happened about this pandemic or COVID-19, we started worrying about its effect on brain. Initially, though, it was very tough to convince the people that it will also have some effect on the brain, because the major symptoms which were very clearly and evident across human population in the world was that the SARS-CoV-2 or the novel coronavirus was affecting the lung tissue. So what came to our support was these post-COVID symptoms or complications, which were fatigue, tiredness, difficulty in breathing, compromised lung function, cardiac events, deep vein thrombosis because of increased coagulopathy in these patients, and then the neurological complications, which were headaches, confusion, unable to concentrate, disturbed sleep, and stroke, higher incidence of stroke. And that tilted a little bit of interest of some neurologists as well as neuroscientists into the understanding the COVID complications. And that brought to the field of brain fog. And this brain fog problem is really huge. People don't understand, but it is actually 40 to 50% of long COVID patients. Not every COVID-19 patient, but those with long COVID patients have reported signs of brain fog or mental confusion, which makes it a significant condition and adds it into the list of common COVID-19 symptoms, the long COVID symptoms. What it does, it impairs your cognitive abilities. You will not be as normal as you were there before in calculations, concentration. Maybe as a researcher, it may take you longer to read the same research paper which you could study or read a similar research paper if you were studying it before. It also causes mental fatigue and confusion. And all this is indicative is that there's something happening in the brain. Either the neurons are not functioning properly, they are not as agile they used to be, or there is some neuronal damage, and that is what needs to be studied. It also has shown as a lack of focus and concentration, memory problems, unclear thoughts, and you may face the difficulty in making up your mind and deciding on one of the most routine things of your life, which you could do very easily before you had the COVID or the SARS-CoV-2 infections. Now, let me try to play this video, and please let me know if you can hear the sound of this, because this clearly shows what a brain fog patient is going through in COVID-19 survivors. A very touchy video which explains exactly what a brain fog patients are going through. So as I said, if you look at these dates over here, in July, this paper was accepted, which we had submitted in April 2020, which was, if you remember, 24th March was the lockdown period. And since our labs were closed, we have nothing else to do, and we were worried about this virus or its effect on brain. We wrote a review on and submitted in April, which got accepted only in July because we had a tough time to explain to people and the reviewers that actually this is possible. And the title we gave to that review was SARS-CoV-2 is more than a respiratory virus and its potential role in early pathogens. So this is what Dr. Charan Singh was referring to in the introduction. So what in this review we had suggested was that SARS-CoV-2 is probably affecting or causing neuron and glial cell necrosis. Okay. It also causes a syndrome which is called as GBS. It is, and these thinkings were based on our knowledge about this virus or the previous sister viruses, and also some of the symptoms which are being were recognized by WHO worldwide, that people having SARS-CoV-2, at least in the first round, were having problems of losing sense of smell, losing sense of taste, and brain lesions, encephalitis, meningitis, hemorrhages and stroke which were being reported. And these are things which we had alarmed the neurologists to be wary of. And people gradually, after the review came up and more patients were reporting to in the hospitals, this review got momentum of attention. Now, later on, in 2021, a much bigger patient population, that was just a review, but a study which was published in Lancet showed that more than in 2 lakh people, what they showed is around 33.62 patients of these COVID-19 patients reported neurological diagnosis, which means that this is an average. But if you look at those who had to go into longer admission times or in ICUs, this percentage was high up to 46 to 62%, which is very, very high, which means that COVID-19 has a neurological consequence. And there were 14 types, which were top types, which were reported in these COVID-19 patients. Now, that debate was that the neurological symptoms which are being reported in brain fog of these patients, is it because of the presence of the virus in the brain? In other words, is the virus itself getting into the brain and then causing this disorder? Or it is just the cytokine storm which is happening in the blood due to the lung infections. And that huge surge of cytokines and chemokines is causing the damage in the brain. And that was debate is still continuing, but now more and more evidences are there which suggest that at least in medulla oblongata and cranial nerves, if you look for the viral proteins of this virus, as such as anti-nucleocapsid or anti-spike protein, which is it has been reported in part of post-mortem tissue. What I'm showing you is a single brain section over here, but if you look at detection of SARS-CoV-2 both by RNA in cryopreserved tissue, formalin-fixed tissue, if you look for nucleocapsid and spike, many tissues have reported. And as more and more post-mortem studies have been done recently, it is more clear that in fact this virus is present in the brain, if not in all, in many of these patients. This has also been supported by another review which came last year in ACS Chemical Neurosciences, which talked about not only single patient studies, but as much as 10 patients, 43 patients, 33 patients from Germany, another 21 patients in Netherlands, 20 brain sections in US, and all these have reported now the presence of virus in the brain. Sorry. I'm sorry. So what all these things are suggesting is that the virus is like in HIV or in Zika virus, SARS-CoV-2 or novel coronavirus is also present in the brain, both as because viral RNA as well as the proteins of this virus are being reported. And that brings you to the question that what are the possible routes of passing of this virus into the brain? Now, again, from our review in April 2020, before there were any studies or post-mortem studies at that time, we were among the first people to suggest that perhaps because it was affecting the sense of smell, we thought olfactory nerve fiber can be one of the ways by which it can cause through the droplet infection, so the mucous membrane infection, it gets access to the olfactory neuron, and it can easily pass into the optic bulb, which was later on actually also proven because in the post-mortem samples, the first presence of the virus was reported from the olfactory bulb, which suggests one of this route to be possible. The other route, of course, is because of the cytokine storm that the blood-brain barrier may open up, the tight integrity of these endothelial cells and the astrocyte end feeds, which Dr. Kakkar was also talking about when she was talking about the availability of curcumin being affected in the brain cells. It is this selectively permeable barrier which protects anything through the blood to get into the brain, and it is supposed to block the virus passing through the brain. But in the terms of endothelial cells, this cytokine storm, these endothelial cells loosen up, the blood-brain barrier opens up, and this virus can get into the brain. And of course, because diarrhea was also reported by WHO and in a lot of patients in the COVID-19 outbreak, we also suggested because there's a gut and brain connection and there's a rich supply in the gut system, the damage to the enteric epithelial cells can actually lead to the leaky gut, which could help in the virus getting into the brain, and then it can affect the nerve cells and nerve tissue. Now, later on, it was also supported by another review, which showed that three major mechanisms, out of which what we had suggested way back in 2020. And this Nature review from neurology did suggest that the inflammatory cells, the microthrombi, which is formed as microhemorrhages, which are also reported in the brain, and of course the virus can get into the brain. So these are the three ways by which the brain can be damaged. But all this was, all theory, because most of the labs were closed, and people wanted to study how CNS manifestations are occurring because of COVID-19. So obviously there are two ways to do it. One is in vitro studies, like the model system what we have of human neural stem cells. Now, since we have neural stem cells, we can make neurons and astrocytes from them, and then put the COVID-19 protein, or the SARS-CoV-2 protein, onto them, and look at which protein out of this virus, which makes 25 proteins or so, which protein is causing the damage to the brain cells. And then obviously we can validate all those in vitro studies by post-mortem samples if we can get them. So a recent approach across the world, including in our lab, has been either using the human-induced pluripotent stem cells-derived neurons, astrocytes, and microglial cells. You put the virus on these cells and find out how these cells are, whether they're infected or not, and if they're infected, what are the kind of damages occurring. Or you can also, if your lab is well equipped and you have the technology, you can develop the cortical organoids, hippocampal organoids, hypothalamic organoids, midbrain organoids, and then try to put the virus there, and that these are considered better than single cells. But if you want to identify the involvement of each cell, may have to go in a single cell preparation. So you would like to have both the model system if possible to do detailed study to understand the mechanisms of SARS-CoV-2 induced neuronal damage, which may be causing the brain fog. So in our lab, what we have started doing, once the labs were open, we looked at all 23 proteins or so, 27 proteins to start with. We started screening them first of all. What we did was a simple live data assay by fluorescence, and then we backed it up with FACS studies, the flow cytometry studies. And what we did, we shortlisted the plasmids. Once we shortlisted out of those 27 plasmids, we took 3 or 4 of them, packaged them in the antivirus, lentiviral packaging, and then we used the lentiviruses to infect the human neurons, which we developed from that. And here, what we're trying to understand, there's no doubt that some of these viral proteins are damaging the neurons or causing death of these neurons. What we are interested to understand, what is the mechanism of that death, whether it's apoptosis, autophagy, necroptosis, paroptosis. In other words, for all possible cell death pathways, we are looking into trying to fish out the markers of that pathway, and then perhaps offer to the field the ways by which this cell death can be prevented in these COVID-19 patients. What we found so far is ORF6, NSP2, NSP8, these three components of the SARS-CoV-2 out of all these is which is causing the maximum cell death of these MCF7 cells. This is breast carcinoma cell line, but was easy to grow and do the flow cytometry, and then we validate the same thing in the human neurons. And what we are also now trying to, we have some handle on, it is the necroptic marker in ORF6 at least, which is going up. So we have now also some understanding which cell death pathway it is following, because we have huge increases in MLKL enzyme with this ORF6 component of the SARS-CoV-2 virus. Now we have also looked into phospho-MLKL and phospho-RIP kinase 3. We did not find any effect in one, but we do see that RIP kinase 3 is being upregulated in the SARS-CoV-2 viral protein ORF6 or ORF6 part of this. We then validated this with human post-mortem samples. Now this, what you're looking at over here is a normal hippocampal region from a human patient, human brain, and a COVID-19 45-year-old female who unfortunately succumbed to the COVID-19 disease, and her brain sections are indicating there's huge cell death which has occurred in that. What we're trying to do also is double labeling to see whether we also can look at the viral protein in vicinity. And then, of course, we are trying to also look into the mechanisms by which the cell death would have occurred. So if I have to summarize this, I would like to say that there's a growing evidence now that SARS-CoV-2 and brain connection is there, but most of the knowledge is mostly based on case studies. And now a lot of studies are happening in vitro and organoid model systems, and the post-mortem studies are also coming very, very handy. Also in our country now, we are fortunate to, at the NIMHANS Brain Bank, Dr. Anita Mahadevan has been able to do some autopsies on these COVID-19 fatalities, and she has collected some brains, and we had access to that with her collaboration. And what we are trying to understand, the detailed mechanistic studies are in process, are definitely very much needed in human brain cells, and which are in progress at the National Brain Research Center. Researchers here are also successfully developed the in vitro model and of molecular basis to understand the molecular basis of SARS-CoV-2 induced neuropathogenesis, and we are identifying which component of the virus is causing the neuronal damage and the mechanisms of neuronal damage as well, so that we can offer some treatment or line of treatment at least. Over the period of years, while we were working, before we were working on SARS-CoV-2, while we were working with HIV and Zika, we have developed this human neural stem cell platform, which is of primary origin, which means these cells have not been tampered with, they have not been immortalized, they are primary cultures. There's a huge advantage of primary cultures over secondary or cell lines. We grow them as monolayer, we grow them as 3D cultures. We also have another source of these neural stem cells from human-induced pluripotent stem cells, iPSCs, which we derive from human blood. We are also engaged in forming co-cultures of human neurons and astrocytes. Again, these are primary cultures, have immense potential for research of human origin cells. And as Tushar also had mentioned, the collaborations are very, very important. And so we have been able to develop a lot of collaborations as well, both international and national, for the use of these stem cell platform for various disorders, including in NBRC and other institutions in India, as well as abroad. With that, I'm really thankful to a lot of people in my lab, very highly talented graduate students and postgraduate student, Dr. Rituparna Chaudhary, who was instrumental in the SARS-CoV-2 work, and of course the other graduate students at different times, which have contributed to this project as well as the other research in my lab. The people at Civil Hospital are also very, very helpful for getting the aborted fetuses, particularly Dr. Sharma, Dr. Singh, and Dr. Thapar, who have helped us with these human fetal tissue. And of course, this is the vibrant group I was talking about. We also had a CV Raman fellow from Africa who spent quite a bit of time with us and has gone back to establish his own lab in South Africa, and he's doing very well. And many of these people are now, Priyanka is now at NIHS, and Chitra is now moving again. He's completed a thesis, and so on. So people are. I'm really grateful to all these people who have helped in at various stages in the lab. Uh, Collaborations were also with my mentor Eugene Major, Avindranath as the clinical director at NINDS and NIH. Funding, as was also mentioned earlier, has been from different sources, and I'm very, very thankful to all the funding sources for their constant support of our research and believing in us. With that, thank you so much for your patient hearing. I hope I am within my time, which was allotted to me. Thank you so much.
Thank you so much, sir, for such an informative talk on the recent issues which we are facing now, even in 2 years. So we have learned a lot from the presentation of the data you have shown from the various review papers, as well as your own research laboratory, sir. And it was nice to see the particular the figure which we have shown in terms of the neurons, I think, along with the post-mortem samples of the COVID-19 patients. And one of the most important points from your own lab is, sir, to study the effect of the HIV virus on the stemness of the NSC, the astrocytes, the glycolysis. And secondly, from the same lab of yours, that the 2D culture of the neurons. So I can remember the same point when I had an opportunity to interact with you and to study the effect of the lopinavir samples then. So these are the most important points from the presentation which we are, which we have done. Even we will try to inculcate if the facility or the time allows. So this is important. So overall, sir, this is an informative and the scientific talk. So and related to the theme of the Ibro, creating sport and culture. Thank you so much, sir, for sparing some time. I know, I think there is a meeting at your end.
Thank you. Thank you.
So,
Always a pleasure.
Sir, there is
Always a pleasure.
one question from my side, if you.
Sure.
Oh, thank you, sir. Sir, as you spoke about the brain fog, like the effect of the COVID, so SARS coronavirus 2. What are your insights on the chemotherapy-induced cognitive impairments that is known as chemo brain versus the brain fog, the effect of virus on the brain?
So you're asking if I.
is also
So your voice was breaking. So what I could understand from your question was that what is the difference between brain fog and chemo brain? Is that the question? Yeah. So delirium is seen in both the cases. Okay, especially those people who are in ICU. Now we still don't know what are the mechanisms of brain fog, and is it because of the the chemokine storm or the cytokine storm what we have following the lung infection, or it is the virus which is getting into the brain. When we look at the post-mortem studies, again, you also agree that that is the end stage of the disease. So these are the most severe cases we are looking into when we look at the post-mortem samples. And that is where the problem lies. In some cases, virus is there. Some cases, it is not there. So what the field is trying to do now is look at the severity of the brain fog or the damage to the brain cells in the postmortem samples and correlate it with the presence of the virus. Is the damage more where the virus is there? Or is it similar whether the virus is there or not there? So these kind of studies which are in progress in our lab as well as in others across the world will give more insights into the role of the virus per se or the viral proteins per se on the brain damage. And because we're looking at both the things, some of the brain damage is also occurring because of the microhemorrhages which are occurring in these brain tissues. Now, similarity of confusion in these things in chemo, brain and brain fog is somewhat similar. But the good part is that some of these patients are also recovering, especially the younger ones from the brain fog. Like we got the sense of smell back within 7 days because fortunately the neurons were not affected, but it was the substanticular or the support cells of the olfactory neurons which were infected. And that again is based on the presence of the ACE2 receptor on the support cell and not on the neuron. So the virus tropism is dependent on the ACE2 receptor. And since brain tissue, particularly neurons, do not have the ACE2 receptors, virus doesn't have any reason to go into the brain, except that the blood-brain barrier is open. So it is so much we have to learn from it as of now. It's still not clear. Any other questions?
Thank you so much, sir,
Oh.
thank you so much, sir,
Always a pleasure and good to
for
see
your
you.
enlightening talks, sir.
Good to see you, Dr. Singh, to do well, and I'm glad you are at a good place and doing well. We'll probably try to come sometime in person there to see your lab. Bye-bye.
Sure.